Any questions before we get rolling today? All right, we got a very busy day ahead of us. Today is Thursday, March 18th, 2021. Today, we will start our discussion of that really, really long physiological process we talked about, deciding how a muscle is going to contract. And that's what we'll be doing and starting in lecture today. And then in lab, we will move on to what we're going to be doing basically for the rest of the labs for the muscular system, focusing on origins, insertions, and actions for the muscles that are on your list. You have three more big assignments that you actually four. I lied, four more big assignments. You have your unit review 10 that is due Thursday. Notice Tuesday the 23rd isn't on here, not because we don't have class, but because there's no assignment due on that day. Although I have noticed we haven't had a daily quiz in a while, so a day like that might be a good day for something like that. But on Thursday, your Unit 10 review next week is going to be due. Then you get a whole week off to master this material and to also work on your PhysioX exercise 2, which, as you remember from the last time you did a PhysioX, isn't hard, it's just time consuming. And this time you have 7 different activities that need to be collected and done all the data and get your lab reports. And so it will take time. So hopefully you can take advantage of the vacation to do that. And then also that week, your Unit 11 review is going to be due, and also a muscle activity handout, which will be graded for correctness, which we'll talk more about when we get to the lab today. All right, so that is the game plan. Any questions on that? I'm sorry, go ahead. Yeah, so exactly. So, and again, you have to save after each lab exam, after each exercise as well. You can't do them all and then try to save it at the end.
Yeah, I learned the hard way.
Well, but so that means you learned the information really, really well. So again, as you remember, it's not hard, it's just time consuming. So make sure you guys focus on that. All right, excellent. So as I mentioned, and again, one of the key things to remember about this is a long, continuous physiological process that we are artificially breaking up into 3 more easily digestible pieces. But one of the important things to remember about that is it is a continuous process. So as we go through this, when we get to the end of part 1, guess what the end of part 1 is?
Is it
First part?
latent
Yeah,
phase?
it's going to be the start of part 2, right? Because it's a continuous process. Normally it goes through continuously. So when we get to an end of one of these 3 steps or these 3 phases that we're going to do, the end of that one part is going to be the beginning of the next part. So when we think of the individual steps, it's something that's important to remember. Now, the first step involves is what is called a communication at the neuromuscular junction. And that is kind of what we are seeing going on up here. Again, when we think of these 3 steps, it is important to think of them in terms of our goal for that step. And the goal for this first step of communication at the neuromuscular junction is to take this information from the language of the nervous system, because remember, this whole thing starts with me deciding I want to contract that muscle. And so I have to take it from the language of the nervous system, which is a neural action potential, and I have to convert it into something that our muscle is going to understand. I need to stimulate the muscle and get that muscle action potential. So our goal of step 1 is going to be to convert our neural action potential into a muscle action potential. Obviously, this is going to require some fancy work to do that properly. And this specialized structure where this occurs, this area of communication that we'll talk about, is known as the neuromuscular junction. So our communication is going to take place at this neuromuscular junction, and our goal is going to be to convert a neural action potential into a muscle action potential. All right. Once that occurs, our muscle action potential is going to spread down the sarcolemma of the cell. And that will be the end of step 1 and also the beginning of step 2. Step 2 is called excitation contraction coupling. What does it mean to couple? These are the easy questions.
Pair
Put
up.
2 things together.
Yeah, to bring together, absolutely. And that's the goal. Notice we have produced a muscle action potential at this point. We have our excitation, but we actually want to start to generate the force. Our goal is going to be to generate force, right? We want to move, we want to contract that muscle, move our body through space. So we need a way to bring these together. And if you remember, we talked about how myosin and actin, they want to interact all the time. Can someone remind me why they don't interact all the time?
Isn't their sensor blocked unless a messenger sends a signal to open it?
You got the right idea. Remember, we had those regulatory proteins that were in the way. So exactly. So if you think about it, to get that contraction to take place, we need to move those regulatory proteins. And in fact, that is going to be the goal of step 2. We are going to move the regulatory proteins. And if we can move those regulatory proteins, then the contraction can begin. Of course, moving those regulatory proteins is going to require something special, something, dare I say, wonky. And so we're going to need to release that calcium from the sarcoplasmic reticulum so that we'll be able to move those regulatory proteins and allow the myosin and the actin to interact. And so that will be our goal of excitation contraction coupling. We are going to use calcium to move our regulatory proteins. Finally, once those regulatory proteins are out of the way, then as you guys were talking about in the last class, we can then get to the actual process of generating tension. And of course, that is going to be our goal of step 3. Our goal of step 3 is to generate the force or the tension, however you want to think of it, that is going to move the muscle, change the shape of the muscle. Right? And that process is what is known as the contractile cycle or the sliding filament theory, like we were talking about last time. Again, as we'll see, these filaments, the myosin and the actin, are going to slide across the surface of each other, which is why it's called the sliding filament theory. I personally tend to like the term contractile cycle because it has that word cycle in it. And what does cycle remind us of?
Goes in a loop.
Yeah, it's a continuous process. So it isn't just A, B, C, D. It's A, B, C, D, A, B, C, D, A, B, C, D in a continuous process. So as we'll see, the contractile cycle is going to be a continuous process that allows us to continue to produce that tension that is going to be necessary. So these are indeed our 3 major steps. You know, we've artificially chopped up this process from decision to contraction into 3 more easily digestible pieces. If there was a 4th process, what would that 4th process be?
The muscle relaxing.
Yeah, once we get, exactly. Once we get the muscle to contract, we don't want it staying contracted forever. If there was a 4th step, we could say that 4th step would be the muscle relaxing.
Я сейчас в классе. Mom, я сейчас тест делаю.
You're still on mic, Meal.
Oh, I'm so sorry.
No worries. The 4th step, if there was a 4th step, would be the relaxation of the muscle. All right. Now, again, we can and we will actually write it out like it could be a 4th step, but there's another way we could think about this as well for all of these. And let's cheat and actually go back to our whiteboard. Because again, it's good to beat this dead horse. Step 1 is communication at the neuromuscular junction. And our goal is to convert a neural action potential into a muscle action potential. Right? That makes sense. We talked about that already. However, as we just mentioned, we can really think about this step in two ways. We can think of it in terms of initiation, where we basically turn it on, and we could think of it in terms of termination, where basically we turn the process off. So rather than thinking of relaxation as a separate fourth step, we could just think of this in terms of you turning the light switch on or you're turning the light switch off, right? They're both same sides of, two sides of the same coin. However, this happens to be a three-sided coin, because if we truly understand a process, then we can understand what happens when we modify the process. If we change something, if we manipulate something, how is that going to affect the process? So really, when we talk about this step 1, communication at the neuromuscular junction, there are really three ways that we can discuss it. We can talk about how we initiate it, we can talk about how we terminate it, or we can talk about how can we modify this communication at the neuromuscular junction. And guess what? It works the same way for the other two as well, sort of. Step 2, anybody remember what step 2 is called?
Contraction.
Close. We need to get to the contraction, but we just have that.
Excitation, contraction, coupling.
There you go. Excitation, contraction, coupling. And anybody remember what the goal of this step was?
To move regulatory proteins.
Excellent. We want to use calcium, oops, to move the regulatory proteins. And of course, again, we can talk about initiation of the process. We can talk about termination of the process. And we can talk about modification of the process. And the same thing goes for step 3. Contractile cycle, or again, you can also use sliding filament theory. Excellent. And our goal here, as we talked about, is to generate the force or the tension to change the shape of the muscle. And again, we can talk about initiation of the process, termination of the process, and modification of the process. And remember, we also talked about, and we'll put it in parentheses as step 4 here, we can take all the terminations together, and that would basically be relaxation of the muscle. So let's actually write it that way. Right. Relaxation of the muscle is to stop. Muscle contraction, but basically it's all the steps of terminating step 1, terminating step 2, and terminating step 3 together. So this is really not new information. It is just organizing the information from before. So notice, if you think about it, This list gives us 10 possible essay questions. And again, remember, as I mentioned, I guarantee you at least 2 will be on your next exam. Now, as it turns out, it's not quite as simple of this. And in reality, and I'll cheat and just do this really small. It's really only going to be 7. We'll see how that is, and I'll explain that when we get to it later. But at least you can see where we come up with the number 10, and then you'll see why we throw some of them out as we work our way along this path. But this is going to be our goal for the next several weeks. Our goal is to understand how to initiate these 3 steps, how to terminate these 3 steps, and to show that we truly understand them, we're going to look at ways that we can modify these steps that really shows that we understand it. And all of these things are possible essay questions that I guarantee at least 2 of will be on the exam. Excellent. Sound like fun? This is the part where you all enthusiastically say, yes, it does. It sounds amazing. Excellent. Glad to hear. Exactly. All right, spectacular. All right. So before we can actually get into those steps, we do need to talk a little bit more anatomy, starting first with the neuromuscular junction. Again, as I mentioned, this neuromuscular junction is a specialized region of communication between our somatic motor nervous system, because remember, we know it's our somatic motor nervous system that controls our skeletal muscle, and the skeletal muscle itself. And again, this is going to be where we produce our muscle action potential. This is going to be where we generate that muscle. I mean, we produce or convert that neural action potential into a muscle action potential. This area of communication, the neuromuscular junction, is a specific type of area of communication known as a synapse. We haven't talked about neurons yet, but let's talk about a little neuron anatomy. And for this, I will, where am I going to do this? We'll cheat and do it here. We'll go back to the whiteboard. All right, clear all of that. A neuron basically has a large centrally located cell body. All right, pretty simple and straightforward. Most cells we know have a big cell body. However, what's different about a neuron is it has processes. Processes is a term that should be vaguely familiar because we just finished talking about bones and how bones can have processes. Well, with our neuron, there are 2 main types of processes, 2 main types of things that stick out. The first of these are short, relatively elaborately branched processes that stick off of the cell body. And a typical neuron has many of them. And these short branched processes are called dendrites. Their job is to receive information so that that information can be processed. But then a neuron also has a single long, and I'll cheat and bend it around this way so it helps me. A single, single, right? Long, unbranched process. And this single, long, unbranched process is called the axon. This axon's job is to carry the neural action potential to the target cell, whatever this neuron is going to communicate with. Here's our target cell. And I do mean that these can be long. If you think about it, No, desmosomes, remember, are the types of cell junctions that lock a cells into place, right? These are just like elaborate branches off of a tree, right? They're branch-like structures that come off of a tree that allow them to receive information from other cells. You have some cell bodies for some of your neurons that are located in the small of your back. And their axons travel all the way down to your big toe. That's about 1 meter in length, right? If you're someone like LeBron James, the small of your back to the tip of your big toe is like 47 feet. So these things can be incredibly long for carrying information long, long distances. And this area of communication at the end, the generic term for it is a synapse. So that area of communication between a neuron and its target cell is called a synapse. Now, to get to that synapse, our axon has to do one more thing. Typically, where it gets near the end, it branches. And as it branches, it forms these structures that are called axon terminals. And it is those axon terminals that typically communicate with our target cell. Now, we now have that basic anatomy and a basic understanding of what a synapse is. So again, a synapse is a generic term. Right, so a synapse is a generic term for the area of communication between a neuron and its target cell. And since that is the generic term, the neuromuscular junction, notice neuromuscular is all one word. Junction obviously is all one word. And the good news is if you're writing this out as an essay question, once you've written this out once, you can abbreviate it NMJ for neuromuscular junction. The neuromuscular junction is a specific type of synapse between the somatic motor neuron and the skeletal muscle cell. So a neuromuscular junction is just one of literally hundreds of types of synapses that are found in the body. So when we look at the anatomy of a neuromuscular junction, we'll see that this neuromuscular junction has 3 main components to it. Let's take a look at this. Let's go ahead and draw this. We'll cheat and start first with, what are we going to start with? We'll start with the neuron first. I like that. And actually, let's start with the muscle cell first. So here, let's go ahead and make it black. Here is our skeletal muscle cell. And we'll cheat and just say that we're looking at a part of it, because we know it's a big, long cylinder shape cell. And as we know, of course, it has this special sarcolemma, which again is the specialized plasma membrane. And remember, we talked about how the plasma membrane of our muscle cell is specialized to produce and propagate, spread, a muscle action potential. Well, we know, like, for instance, the sarcolemma has transverse tubules, folds in it that travel into the muscle cell. But there is another specialization. There is a specialization of the muscle cell that is a part of the neuromuscular junction. And it is called the motor end plate. One of the specializations to the motor end plate is that it has a very large surface area. Oops. So what happens is there are many folds in the plasma membrane in this area, large number of invaginations, and these invaginations give it a very large surface area. There is also a neural component, and let's draw the neural component of this as well. As we just finished seeing, we know that there's going to be an axon terminal that is going to travel down. And what I didn't show you is the end of the axon terminal. At the end of the axon terminal is a big enlargement. And this big enlargement is the specialized part of the neuron be specific specialization of the neuron that is part of the neuromuscular junction. And it is called the synaptic end bulb. Now, one of the things I will warn you is that there are many names for the synaptic end bulb. Over a dozen names for the synaptic end bulb. It can be referred to as the terminal end bulbs, the terminal boutons, synaptic buttons. There's all sorts of different terms. I'm going to use synaptic end bulb. You may use any appropriate term. So if you've learned about this anywhere else, you're welcome to use any of those. But the key to it is that it has specializations as well. While that neuron sends a big, huge electrical signal the whole length of the neuron, it turns out that most neurons communicate to their target cells by releasing a chemical signal. Anyone know what the name of that chemical signal, the generic term for the chemical signal that a neuron releases to communicate with a target cell would be? No neuroanatomy fans out there?
Neurotransmitter?
Absolutely. Neurotransmitter. Absolutely. Over 100 different neurotransmitters have been discovered. However, here in the synaptic end bulb, in our neuromuscular junction, there is one specific neurotransmitter that is used. And in this case, the specific neurotransmitter used is a neurotransmitter called acetylcholine, which, again, in an essay, once you've written out once, you can abbreviate ACH. So inside of this synaptic end bulb, there are a massive number. This is way too big. There we go. There are all these vesicles containing acetylcholine, a chemical signal used for communication. And let's draw one more and write ACH in it to remind ourselves of what's in here. Excellent. So when our neuron releases this acetylcholine, it is going to stimulate our muscle cell. Notice our muscle cell has a nice large surface area to it that it can receive that chemical signal. However, to receive that chemical signal, it has to have some special receptors on this motor end plate as well. Vesicles. Remember, vesicles, as we talked about, are just small pieces of plasma membrane that can contain proteins and stuff like that inside of it. So we have these vesicles containing a massive amount of acetylcholine that they will be able to release. So on this huge, large surface area, this big invaginated motor end plate, what we have here are some special receptors, some channels. And these are going to be chemically gated channels. So this is a channel whose door is unlocked by the acetylcholine. So acetylcholine unlocks the door and opens the door up. And once that door is opened, it is cations that pass through it. Someone remind me again what a cation is?
Positively
It has a
charged.
positive.
There you go. It's a positively charged ion. Absolutely. So it's a big fancy term, chemically gated cation channel. And you will also sometimes see the term ligand gated channels, but it means the same thing. It just means that there's a chemical that opens it up. And again, remember what this means. That means that there is a chemical, in this case, acetylcholine, that opens the channel. But it's not the acetylcholine that passes through it. Instead, it is positively charged ions that either enter or exit the cell through the open channel. And just out of curiosity, does anybody remember what the two most common cations in the body are?
Sodium and calcium.
Sodium is definitely one of them. What was the other one?
Oh, potassium.
Potassium.
Potassium, absolutely. And so if we open this channel and let sodium move, does anybody remember which way sodium would move? Would sodium move into the cell or would sodium move out of the cell?
Into.
Yep. And potassium, which way would it move? Out. There you go. Excellent. So when that chemical binds to this receptor, it's going to open it up, and sodium is going to rush into the cell, and potassium is going to rush out. All right. Now, notice with my synaptic end bulb, do I necessarily want it to be releasing acetylcholine all the time?
No.
No.
No, if I was releasing acetylcholine all the time, the muscle would be contracting all the time, and that would be a bad thing. So I need a special way, dare I say even a wonky way, to tell this synaptic end bulb to release acetylcholine. And what would a good signal for that be? What makes cells do wonky things?
Calcium.
Calcium.
Calcium, absolutely. So over here on my synaptic end bulb, I have some additional channels, some different channels. Oops, this isn't gonna work right. There we go, put that one there, and that one there, and this one here, and this one here. And these channels are what are known as voltage gated calcium channels. Let's break that down. Let's start easy. And here, we'll put it up here. Voltage gated calcium channels. What moves through the channel when the channel is open?
Calcium ions.
Calcium is going to move. And which way do you think calcium moves? Into the cell or out of the cell?
Into.
Yeah. So when the channel open, calcium moves into the cell. Excellent. But this is a gated channel. That means that it can be opened and closed. So what is the key, do you think, to unlock a voltage gated calcium channel?
Is it adrenaline?
Not a bad guess, but remember the other one was chemically gated, and it turned out it was a chemical that opened it. This is voltage gated,
Electrical signal?
an electrical signal. Remember, one of the things we've talked about is cells have a resting membrane potential. Does anybody remember what we said the resting membrane potential of a cell was?
-70 millivolts.
Negative 70 millivolts. Excellent. So it already has a charge as it's sitting here. If we can change the membrane potential, we can open this channel. And that's what's going to happen. The key to unlock it, to open the channel, we have to change the membrane potential of the cell. So if we can change the membrane potential of the cell, the channel will open. And when the channel opens, calcium will move through it. Nope, I did not give myself enough room to write that, so I'll sneak it up there. Oh, great question. So yes and no. So one potential cause for cramping can be an imbalance in ions. Absolutely. However, notice it isn't just calcium. We have calcium, sodium, and potassium all at work here. So notice imbalances of any of these can actually cause potential inappropriately firing of the muscle, because that's really what a cramp is. A cramp is when the muscle fires inappropriately. And ionic imbalances can be one of the things that causes it. It's one of the reasons why, again, for those endurance athletes, one of the things that they swear by is bananas, right? Get a lot of potassium and other stuff like that from things along those lines. They drink their chocolate milk after to get their calcium, right? Things along those lines. To stay hydrated, they don't just drink water, but they'll take a salt tablet with that water to maintain their sodium levels. So absolutely, ionic imbalances are one of the things, not the only thing that can cause cramping, but one of the major causes of cramping. Absolutely. All right. So notice we have these two specializations, the synaptic end bulb and the motor end plate. And the synaptic end bulb has a couple specializations to it. It has vesicles containing acetylcholine and voltage-gated calcium channels. There's one other thing in here as well that there's a lot of. It's these kind of kidney bean shaped structures that have a double membrane inside of them. What might that sound an awful lot like?
Mitochondria?
Mitochondria, because it turns out this process of releasing the neurotransmitter requires a tremendous amount of ATP. So we need a lot of mitochondria in here to produce ATP as well. So we have these specializations to our synaptic end bulb. We have our specializations to the motor end plate, this massive invagination and these chemically gated cation channels. And one last thing, I think I may have said it, I don't remember if I said it out loud or not. I said it in my head. One important thing to remember is that each skeletal muscle cell has one and only one neuromuscular junction or motor end plate. So again, only one neuron talks to each individual muscle cell. Well, we'll get to that. We are going to absolutely describe the process. However, we're not done describing the synapse yet either, because notice, not too subtly, I have pointed out here that while there's all this good stuff going on, you will notice that the synaptic end bulb and the motor end plate don't actually touch. There is space in between the two. And that space in between the two structures is what is called the synaptic cleft. So our synaptic cleft is the space between these two. They come close, but they don't actually touch. And the space in between is the synaptic cleft. The synaptic cleft is filled with interstitial fluid. This is important because, as we know, what's inside the cell is different from what's outside the cell. We know, for instance, there is lots of potassium inside of a cell, and there is very little potassium outside the cell. Lots of sodium outside the cell. Oops. Lots of calcium outside the cell. We know all of those characteristics that we've talked about before, different from what's on the inside. But there's one other special thing that we find in here as well. There is a special enzyme that breaks down our neurotransmitter. And I'm just going to abbreviate neurotransmitter like that because of the interest of space, because I'm going to need some room to.
Open up that little box that you put below the little potassium.
I'm sorry, say that again.
Move up the box because I couldn't see the somatic thing you typed on the bottom of the.
Oh, you can't see the. Oh, okay. Move that up.
That one, yeah, thank you.
Can you see lots of potassium?
Yeah, I see a lot of it.
All right, excellent. Okay, perfect. So now you can see everything? Yes? All right, sounds, I'm going to assume means yes. Excellent. So the last specialization we have to talk about is a specialization found here inside of our synaptic cleft. And I think I'm going to need the smaller font to sneak this in. A special enzyme that breaks down our acetylcholine. And that special, maybe I'll sneak it up here. That special enzyme that breaks down acetylcholine is called acetylcholinesterase. And again, the good news on an exam is you only have to write that out once in an essay question, and then it can be abbreviated ACHE. So these are the components of our neuromuscular junction. Let's dive back to the lecture and take a look at this. Notice here with this really simple illustration, we can see the invagination. We can see the axon terminals. We can see the synaptic end bulb where it's connecting with it. Although we'll take a little bit of a closer look, and here we see that here. So notice here, we see those specializations that we've talked about. Here we have our synaptic end bulb. Notice it contains the vesicles containing our acetylcholine, voltage-gated calcium channels, and lots of mitochondria to produce ATP. Again, that is a specialization of the neuron, of the axon. Here we see the motor end plate with its large invaginations, increasing the surface area, and lots of chemically gated cation channels, channels that our chemical acetylcholine binds to and opens up so that sodium and potassium can move into and out of the cells. And again, they do not touch. There is a space in between, and that space in between is the synaptic cleft. All right. So our goal here is our synaptic end bulb is going to release that neurotransmitter. It diffuses across that synaptic cleft, binds to our motor end plate, and is going to hopefully generate our muscle action potential. Again, neurotransmitter is the generic term for this chemical signal, but here at the neuromuscular junction, there is one and only one neurotransmitter we use, and that is acetylcholine. So we've got it stored in the vesicles, and the motor end plate then has those receptors for the acetylcholine. That is going to allow the ions to flow in. All right. Questions on that? All right, let's come back here to this picture then. This is the basic anatomy. This is our neuromuscular junction. And now that we have the anatomy of our neuromuscular junction, what we can actually do is we can actually talk about the process by which this occurs. All right. Now, one thing that I want to remind you of, I should have set this up ahead of time, but I forgot about it. I always forget about it. But let's do this. When we're dealing with large physiological processes that can be sometimes confusing, there are some huge resources that can help you to be successful with this, that hopefully you're taking advantage of. And if you're not, then I want to strongly encourage you to do so. And again, the place where you find that is here in your Mastering A&P. And typically one of the easiest ways to find all of the available resources is to do it by chapter. So notice right now we are talking about muscle and muscle tissue. So if I click on that muscle and muscle tissue, not only, I don't know what I said, I wasn't paying attention. Yes, if you're not taking advantage of it, then shame on you. Excellent. Who knows what I was saying? I wasn't paying attention. Excellent. There are practice quiz questions, there are practice tests you can take, things along those lines, things to the textbook. But in particular, the things that I want to focus on for you are these things right here. The first is the A&P flicks. These are high resolution animations that show this material. In fact, if we click right here, and I have volume turned up all the way. Can I make that big?
Nerve impulses, also known as action potentials.
Can you guys hear that?
Yeah.
Okay.
Travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of the skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels.
Or chemically gated.
Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma.
And there you go. Just that easily, we have seen, and we will together with our working together describe communication, initiation of communication at the neuromuscular junction. We have just that easily seen that. So again, it's a great resource, a great tool to use if you're not using it already. The other thing that are in there are these interactive physiologies. These interactive physiologies are tutorials, walkthrough tutorials that can show many of the processes and many of the steps involved in this as well. So some of them include videos, some of them include, there are also labeling activities and things like that that you can see, flashcard makers, more video tutors that you can look at. There are a lot of tremendous resources. An MP3 tutor for the events at the neuromuscular junction for the sliding filament theory. Again, great resources here that if you're not taking advantage of, I strongly encourage you to take advantage of them, because again, it can, some of these concepts can be challenging. And so having these different modalities to be able to learn and make sense of them is definitely something that hopefully will help you to be successful. All right. Questions on that?
I have a question.
Yes.
One of the muscle functions we said it was producing action potential, but doesn't it really like get that potential from neurons? Does it produce, or it gets it from neurons?
So notice, and that's one of the keys where they don't touch, right? This one produces a big electrical signal, the neural action potential that spreads down here. And if we had them hardwired together, where this directly connected to that, then yes, the electrical signal would go straight to the skeletal muscle and it would just spread to the skeletal muscle. And that would be awesome. But it also wouldn't be, because while it's convenient that these things work together, that the neuron tells the muscle to contract, right? And we can take advantage of that. In fact, your doctor takes advantage of that when you're sitting on his table and he takes that hammer and he hits you in the knee. And when he hits you in the knee with that hammer, what happens?
Your knee moves up.
Anyone had their reflexes checked?
Yeah.
What happens when he hits you?
You kick your leg up.
Yeah, you kick your leg up. Absolutely. Right. So you kick your leg up. All right. Showing that the communication between your nervous system and your muscular system is working. But what if he goes to hit your knee again a second time? And before he does that, he takes your newborn baby and sets it on the floor right next to your foot. When he hits your knee, is your leg going to kick out and are you going to kick your newborn baby?
No.
Hopefully.
No. You will override that reflex and your leg won't kick out, right? If you don't have newborn baby, then maybe he puts a tiger that's sleeping there. Are you going to kick the sleeping tiger? No, right? So we want to be able to modify it. And so what happens is by releasing a chemical signal, and then if the signal's big enough, generating an action potential here, that allows modification, right? Hardwiring has lots of good resources. Your house takes advantage of it. I've got a light switch on the wall over there that when I put it up, the light goes on. When I put it off, the light goes off. And I can flip it all day long. And even if I sat there flipping it all day long, at no point is my garage door going to open or the garbage disposal downstairs going to turn on or anything like that. Having things hardwired definitely has its uses, but not here. Here we want to be able to modify the signal. And so here, absolutely, this neural action potential causes the release of chemicals, and those chemicals generate an action potential in that skeletal muscle cell. So we really are converting. It's not just spreading one to the other. We're converting that neural action potential into a muscle action potential.
Okay, that makes sense. Thank you.
Yep. And again, I will walk you through all the steps of it. I know that video went through it quickly. Trust me, we will go through it with excruciating detail to make sure you understand it. And not only that, we are going to figure out how to initiate it. We are going to figure out how to terminate it. And to make sure that we understand it, we are going to modify it. That does sound like a lot of work, though. And since it is a little bit early, yep, this is exactly what we're going to do. We're going to convert that NAP to a map. Absolutely. Even though it's a little bit early, we've got a lot of material to cover. So let's go ahead and take our first break here. Let's take a 15-minute break, come back at 1:10. And at 1:10, we will pick up the lecture from here. I'm going to need to do some writing on the board and make sure that we have it all set up. And we will restart and go through this whole process. Step one, everything you wanted to know and more about communication at the neuromuscular junction. All right, so any other questions before we take our break? All right, then I will see you guys in 15 minutes. All righty. Let's go ahead and get started. So, like I said,
You
our
want
goal.
to praise us for our artwork.
Yes.
So we praise you all the time.
Exactly. Yeah. So if you want to, I'll print it out and put it on my refrigerator if you guys want to add to these. All right, but let's go ahead and get started. All right. So our goal now is to fully understand this process of step one, communication at the neuromuscular junction. And someone remind me again what our goal of step one, communication of the neuromuscular junction is.
That's to convert the neural action potential to the muscle action potential.
Exactly. And I'll make that a little bit smaller so that it fits. There you go. Our goal is to convert the neural action potential into a muscle action potential. And of course, this curves here at what we have half drawn as our neuromuscular junction. Now, our neuromuscular junction, of course, is made of 3 components. This first component right here, of course, is our synaptic end bulb. And if I remember correctly, there were 3 specializations to the synaptic end bulb. Does anybody remember what any of them were?
Would it be like the vessels like in this acetyl-CoA?
Absolutely. So one of them absolutely are those vesicles containing our neurotransmitter. So we have, I'm sorry?
In the mitochondria too?
Yep. We have the vesicles of acetylcholine. Excellent. We have lots of mitochondria. And what was the third specialization?
The axon terminal?
No, remember the axon terminal was just a branch of the axon that was coming down. So we have our axon.
The calcium channels.
Yep, exactly. And what they were gated channels. So what opened and closed them?
Voltage.
Yeah. So voltage gated calcium channels. Excellent. So let's draw all those things. Well, let's draw half of those things, 2/3 of those things.
If you need help drawing, just let us know.
I appreciate how excited you guys are about the prospect now, but I think we'll I'll try to handle it for now, but feel free to add later.
I was totally joking.
All right. Although I will warn you, the one thing about that, and again, I actually found this out in a meeting I was in. If you guys annotate somebody else's stuff, it does show the name of the person who does it. So they do know it's you.
Yeah.
All right, excellent. So there's our vesicles containing the acetylcholine. And I'm going to go ahead and put a couple voltage gated calcium channels up here as well. I'm not going to bother drawing the mitochondria. We know what mitochondria do, but we'll focus on the rest of these things for that way. Excellent. So that is the first part there. Our second part is our motor end plate. It is the specialization of our muscle cell. And what were the big specializations to the motor end plate? Well, I've already got one of them drawn there. What is it?
Chemically gated channels.
Well, you're right. Those are definitely there. Those aren't the ones I've drawn yet, though, but you're absolutely right. I do need to draw those. That is going to need to be there. But what is special about the motor end plate that we can see? Say again?
The invagination.
Yeah, exactly. It has these massive invaginations that increase surface area. It also gives little nooks and crannies for that acetylcholine to stay locked in this position so it can have a larger effect. But you guys are also right. We also need those chemically gated cation channels. And I'll draw a couple of those real simply as well. So there's one, and there's one, and there's one. Excellent. Lastly, we have the space in between. This space in between is called what?
I have a quick question about the space in between.
Yes.
What is it associated with? Is it part of the synaptic end bulb? Is it motor end plate, or is it neither? Like, where does that tissue come from?
But so you got to remember, it's this, the neuron and the muscle are surrounded by connective tissues and other things. So this is just interstitial space. It's neither, right? If I have my two hands and they're close together and not touching, the space in between is not a part of my left hand and not a part of my right hand. They are the space in between it. So that's why this is just filled. It's just space that's filled with interstitial fluid. So this synaptic cleft,
Okay, thank you.
is merely a space that is filled with interstitial fluid. All right. So again, it's just that space in between. Now, again, because it's interstitial fluid, it has things like large amounts of sodium, large amounts of chloride, large amounts of calcium, small amounts of potassium, and all those things that we know are outside of cells. But remember, also in this space, we are going to have that special enzyme. And that special enzyme is acetylcholine esterase. And we'll draw that as just a simple little Pac-Man whose job it is to gobble up those acetylcholine when they're released into the synaptic cleft. All right. So there you go. Just that simply, just that easily, we have identified the anatomy of our neuromuscular junction, the three components, the synaptic end bulb, the synaptic cleft, and the motor end plate. And we've talked about all the specializations of all of them. All right. Questions on this? Again, this is where it starts getting important. If it's not,
I have a question.
Yeah, go ahead.
Okay, so acetylcholine esterase, does it put those, it captures those messenger chemicals, whatever they are. What does it do with them? Does it recycle it back to the synaptic end bulb?
It breaks it. When it finds acetylcholine, it breaks it into an acetyl group and a choline group.
Oh,
so if I take your key and break it, is it going to work in that lock anymore?
No.
No. And so there you go.
Okay.
So that, as we'll see, is going to play an important role on the relaxing of the muscle when we get to that point. All right, excellent. Ariane, I'm glad to hear it's coming together. Again, this all is going to build on each other. So this is the point where it's important, if this doesn't make sense, to make sure you say something, make sure you speak up, because it's just going to get worse. Because what we're going to do is we're going to keep layering more and more and more on top of this. All right, so here in black, we have the basic anatomy that we understand from that. So now our goal is going to be, and let's switch to blue because blue is a good color for this, the process of initiation. Right? This is where we turn it on. So this is the point where I get a muscle to contract. And when I do that, where does it begin? If I'm going to get my hand to contract, to flex like this, my fingers to flex, where does that process begin?
In your brain?
Yeah, exactly. It starts with the decision. Make the decision. When we make the decision that I want to contract that muscle, because remember, we're talking about skeletal muscle, and skeletal muscle is voluntary. We make that decision. And when we make that decision, we generate a neural action potential. Now, I don't remember if we've talked about this. I think we talked about it when we talked about a muscle action potential. But a neural action potential is a big positive signal. It's basically a massive wave. No, this is all step one. Step one is communication at the neuromuscular junction, and we have not communicated at the neuromuscular junction yet. All we've seen is the anatomy of it. We haven't seen how it works.
Oh, okay.
So, in fact, this is the very this is now the second step of part one. We make the decision, we generate a neural action potential.
So basically you were just going into depth of like how it all comes together, like the neuromuscular junction.
Exactly how this works, and I'm giving you exactly what I want when I ask you this question on the exam.
Okay.
So guess what? If on your exam, question one is describe, or first essay question is describe the process of initiation of communication at the neuromuscular junction. Well, the first two things you're going to say is that it starts with making a decision to contract the muscle. And then when we make that decision, we generate a neural action potential, which is a big positive signal. All right. And then that well, okay, the swatting a piece of garbage that's flying towards your face is more protective and things along that way. The catching something that's falling and breakable, those are learned behaviors, right? If you drop, an ancient Ming vase next to a newborn baby, that newborn baby isn't going to reach out and grab it. However, if you touch the cheek of a newborn baby, what is that baby going to do?
Probably smile.
Maybe flinch.
No, none of those things. None of you have had a newborn baby?
It's going to turn its head the other way.
Yeah, now it's going to turn its head the other way. It's going to turn its head towards it.
Yeah, it's going to
try to latch on. It's going to try to eat. Exactly. It's going to turn and try to eat, right? These things are called reflexes, right? And some reflexes we're born with, like that latching on reflex, where they'll turn and try to latch on to whatever you touch them with. Some of them are, things that we learn, right? When you're driving in a car, and you've got a passenger in the passenger seat and a ball rolls out in the street in front of you, you do two things. You slam on the brakes and you reach out your arm to protect the person next to you, right? Newborn babies don't do that. You put a newborn baby in the driver's seat of a car, roll a ball in front of them. They don't, right, reach out to protect the person next to them. These are things that we learn. All right, excellent. So we produce this big positive signal, this neural action potential. And again, because I've written out neural action potential once, I can start abbreviating it. Our neural action potential spreads down the axon to the synaptic end bulb. And since I've written out synaptic end bulb down here, I will just abbreviate it this way. Now, this is a big positive signal. So as it spreads, it makes the cell more positive. And we have a fancy name for it when a cell becomes more positive. Does anybody know what that term is?
Polarization.
Yeah. When a cell gets more positive, the cell depolarizes. Or more specifically, let's do it this way, because right now we're in the axon. The axon depolarizes. All right, so we have that depolarization. That big depolarization then reaches our synaptic end bulb. And it's a big positive signal. Someone remind me again what the resting membrane potential of a cell is?
Negative 70.
70.
Yeah, resting membrane potential equals negative 70 millivolts. So if I've got this big depolarization coming into my synaptic end bulb, oh, that's way too big. There, blue. This big depolarization coming into it, it's going to make my It's going to make my synaptic embolum more positive. Okay. Now, notice we have these voltage-gated calcium channels. And what did we say opened a voltage-gated calcium channel?
The potential gradient.
Yeah, changing the membrane potential, absolutely. So what do you think? Getting the cell from -70 to -69, do you think that's going to be enough to get those channels to open?
No.
No.
What about -68 or -67?
No.
Probably not. But is there a magic number that we need to get to?
Yeah.
Yes.
Yeah, exactly. And so that's the key. If this depolarization is big enough, it will get, actually, let me stick this up here and I'm going to start writing smaller so that I have more room for all of this. If the depolarization is big enough, and the way we know it's big enough is that it reaches that magical number. And it turns out that magical number is called threshold. So if the depolarization is big enough to reach threshold, then the voltage gated, and I'm going to abbreviate that VG, CA, voltage gated calcium channels will open. Now, I will tell you right now that threshold is usually around -60 millivolts. So it's about a 10 millivolt change. And the average action potential is about a 100 millivolt change. So notice that action potential coming down is absolutely going to be big enough to get us to reach threshold and open these voltage gated calcium channels. So our voltage gated calcium channel opens. And when it's opened, what's going to happen?
Calcium comes in.
Absolutely. Calcium is going to enter the cell. We're going to get a massive flow of calcium into the cell. Now, normally calcium doesn't, normally cells don't like calcium being inside of them. So one of the things that most cells have is they have a calcium pump. This calcium pump uses ATP, and it uses this ATP to kick calcium out of the cell. Right? Kind of like if you're in a boat, many people when they're in the boat, especially if they have smaller boats, they have a little hand pump. So if a little bit of water gets into the boat, they can use that hand pump to pump the water out of the boat. That make sense? Again, this is where I take stunned silences as yeses. So voltage gated calcium channel. So our voltage gated calcium channel opens and it rushes in. Excellent. So you've got that hand pump to get the water out of your boat. But what if I drill 75 holes in the bottom of your boat? Is that little hand pump going to be enough to keep all the water out?
No.
No. And that's exactly what happens here. We have a massive amount of calcium coming in. Yes, some gets pumped out, but most of the calcium stays in. And when most of it stays in, we get an increase of calcium inside the cell. So we have an increase in the concentration of calcium inside the synaptic embolum. And as we've mentioned numerous times so far, and we'll continue to mention, calcium makes cells do wonky things. In this case, calcium triggers exocytosis. Now, again, notice the key word there. It triggers exocytosis. Calcium doesn't come to the vesicle, grab the vesicle's hand, and bring it to the plasma membrane. In fact, there are 25 different proteins that are involved in the process of bringing, this vesicle to the plasma membrane. And when it gets to the plasma membrane, it binds to the plasma membrane. And of course, we got to open up our plasma membrane. And that allows it to release its acetylcholine by exocytosis. So again, there are 25 proteins involved with that process. It is an amazingly elaborate process, which I'm not going to require you to need to worry about. But the domino that starts the process, that triggers all of those to do their job, is calcium. So when there's calcium inside of the synaptic end bulb, then our acetylcholine is released via exocytosis into the synaptic cleft. All right. Let's cheat and move that there. Perfect. Questions on that?
I have a question.
Yes, this is all step one. We haven't even, yes, and we're not even halfway through step one yet. Yes.
So those vesicles are there even if the cell is on resting membrane potential?
Yeah, they're inside of it all the time. They store all of these chemicals in there for communication to release when it's time.
Okay.
Yeah. And again, way back, hold on, way back at the cell body, remember we have the Golgi apparatus and the rough endoplasmic reticulum. We make all those proteins, bundle them up into the vesicle, and then ship them down here to the synaptic end bulb so that they can be used for communication. Yeah, someone else had a question?
Yeah, I had a question. So, like, is there like an unending supply of those vesicles with ACH in it, or do muscles that get used a lot have more and muscles that get used rarely have less?
So yes, muscles that get used more will typically store more acetylcholine in them. And think of it this way. Can you contract a muscle forever?
No, but I can
No.
go a long time even without practice until, you know.
True. Yeah,
sort
that
of
we do
stay.
have some, there are some muscles that are fatigue resistant and can be, sustained for a period of time. But theoretically, might it be possible to use it so much that we could use up all of the acetylcholine? Potentially, yes. It would take an incredibly massive, prolonged excitation to do that. And so, again, under normal biological conditions, that doesn't occur. But definitely, it can be depleted where it weakens into the strength that it can produce. Yes, absolutely. And we will, once we start building back up, we'll talk about muscle fatigue. All right. Excellent. Yeah, we're talking about this happening in fractions of a second. Absolutely. All right. So we have released our acetylcholine into the synaptic cleft. And is it just going to stay there huddled up against the wall of the synaptic end bulb when that occurs?
No.
No. So our acetylcholine diffuses across the synaptic cleft to the motor end plate. Oops. So the acetylcholine diffuses across the synaptic cleft to the motor end plate. And when it gets there, it binds to our chemically gated cation channel. Now, again, notice when this acetylcholine is released into the synaptic cleft, is some of it going to get gobbled up by that enzyme that's there? Yeah, but we're releasing a lot more then are able to be gobbled up. So again, there's going to be enough in here to make it across to the other side and bind to the motor end plate. And that's our goal. Our goal here is to bind the acetylcholine to the chemically gated, and I'm just going to abbreviate that CG, chemically gated cation channel. And when it binds to the chemically gated cation channel, the channel opens. Now, when the channel opens, as we know, it's a cation channel, and sodium is going to move. And which way did we say sodium was going to move?
Into.
Sodium enters the cell. Excellent. And we said potassium is going to go what way?
Out.
Excellent. And notice these are both positive ions. So if one sodium comes in, and one potassium goes out, have we changed the membrane potential of our motor end plate at all? One positive thing leaves, one positive thing comes in. Is that going to change it from being negative 70 millivolts? No, that wouldn't cause a change. However, it turns out it is not equal amounts of these that move. As it turns out, there is a lot of sodium that enters into the cell and only a little bit of potassium that comes in. So both sodium and potassium do move, but it is a massive amount of sodium coming in and only a little bit of potassium that is leaving. So notice we have more positive coming in than negative. So notice the net effect is that our motor end plate gets more positive. And wasn't there a fancy word for getting more positive? What was the fancy word? Depolarization, exactly. Our motor end plate depolarizes. And here's the key. And if the depolarization is big enough, and how do you think we would know if the depolarization was big enough?
It reaches a threshold.
Exactly, it's big enough to reach threshold. Then we generate a muscle action potential at the motor end plate. So we're going to produce a big, huge electrical wave, that big, huge 100 millivolt change. Muscle action potential. And once we generate that, it will then spreads down the sarcolemma, because we know our sarcolemma is specialized for production and propagation of the action potential, so it will spread down the sarcolemma. So, Aravec, to answer your question, sodium potassium pumps help to reestablish this. Notice this kicks, so this allows sodium in, this allows potassium to leave, and we don't necessarily want them now. We want to be able to kick that sodium back out. We want to be able to bring that potassium back in. So you are correct. This cell absolutely positively is going to have some of those sodium potassium ATPases. And remember, those sodium potassium ATPases move how many sodium?
I think 2.
3.
3 to 1.
3, yep, 3 sodium out of the cell and 2 potassium into the cell. So yeah, so yes, this pump is going to be here, but again, if we're opening all these channels and massive amount of sodium's coming in, it doesn't matter what this pump is doing because there's this massive influx and we are going to get that big, huge depolarization. And if it's big enough, we get a muscle action potential, and our muscle action potential is generated. And that is step one, initiation of communication at the neuromuscular junction. All right, I want to do two things. The first thing I want to do is go through this again. I have done an amazing job of drawing this on the board, but let's take a look at the pretty pictures from your textbook and remind us about what's going on here. We'll go through it a second time, because again, I think it's always important to have this repetition for it. So again, the process begins with the decision. We decide that we're going to contract the muscle because this is a voluntary action, and we generate a neural action potential in our brain. It is a big positive signal that causes a depolarization of our neuron. And that big depolarization spreads down the axon terminal to the synaptic end bulb. One of the important things to remember when you're going through these processes is to think of what is the immediate effect. The immediate effect of that action potential reaching the synaptic end bulb is it brings the synaptic end bulb to threshold. And when we reach threshold, that causes something to occur. Our voltage-gated calcium channels open. What's the immediate effect of the voltage-gated calcium channels opening?
The calcium comes in the cell.
Yeah, calcium enters into the cell. And when calcium's in the cell, that triggers that big, huge cascade effect using those 25 proteins that cause the vesicles to come to the plasma membrane and release their contents via exocytosis into the synaptic cleft. Once that acetylcholine is in the synaptic cleft, it diffuses across the synaptic cleft and binds to our ligand-gated or chemically gated channels on the motor end plate. Yes, Yulia?
Yeah, I wasn't sure if Eric's question got answered. All of this happens without the use of ATP, right?
Yes, although again, ATP is being used for these pumps, but they're not as important for this part of the process. That is correct.
Okay, so it just kind of happens on its own.
Well, it doesn't happen on its own. You're generating an action potential. That action potential is spreading. We're triggering all of these events. Exocytosis requires ATP. So, I mean, this is an active process. This is something that you're deciding to do and actively causing to occur. So it's not like this is just occurring randomly, right? You don't just have random spasms of your muscles. This is something that is generated and directed, but this is how it works. Yes.
Okay, thank you.
Trust me, when we get to the contraction, we'll use all the ATP you ever wanted to and more, okay? But we're not using ATP so much for this parts. All right. That acetylcholine binds to the chemically gated channels on the motor end plate. Here we see it up close. And again, it is a cation channel, which means that both sodium can come in and potassium can come out. But remember, it's not equal amounts. Only a little bit of the potassium leaves, but a whole lot of the sodium comes in. So more positive ions are coming in than positive ions are leaving. The net effect of that is that it depolarizes the cell. And if that depolarization is big enough to reach threshold, we generate a muscle action potential. All right. Questions on that?
Yeah, where's acetylcholine esterase located in again?
In the synaptic cleft.
So it just floats around?
Yep. Same way sodium and potassium and amino acids and all sorts of other stuff are floating around inside your interstitial fluid. Yep. Just hanging out there in the,
okay.
just chilling in the synaptic cleft. All right. There's one other really important point I want to make about this. Again, we are 3 exams in, so I feel I shouldn't have to say this, but in grading the exams, I feel I do need to say this. One of the things that is going to maximize your points on this exam, on all the exams, is to be specific, is to give details. Notice, I can describe this process to you. We make a decision to contract a muscle. My neuron releases acetylcholine. Sodium enters my muscle cell, and I generate a muscle action potential. What I just said there is 100% correct. But it lacked all of the details. And it is the details that are going to get you the points on the exam. So like I talked about, when you're describing this process, think of what is the immediate effect of that, right? So instead, I can also describe it this way, which is 100% correct. I decide to contract a muscle and I generate a neural action potential. That neural action potential, which is a big positive signal, spreads down the axon to the synaptic end bulb. That neural action potential depolarizes the synaptic end bulb to threshold, which opens voltage-gated calcium channels. When the voltage-gated calcium channels open, calcium rushes into the synaptic end bulb. That calcium triggers vesicles containing acetylcholine to bind to the plasma membrane and release that acetylcholine via exocytosis. That acetylcholine is exocytosis into the synaptic cleft. It spreads across the synaptic cleft to the motor end plate, where it binds to chemically gated cation channels. The acetylcholine opens the chemically gated cation channels. A large amount of sodium enters the cell. A small amount of potassium leaves the cell. The net effect of that is the motor end plate depolarizes. And if that depolarization is big enough to reach threshold, we generate a muscle action potential and it spreads down the sarcolemma. Right? Both of those descriptions were 100% correct, but one had all the details, one had all the specifications, one had every intimate step of the process. And so one of those was 100% correct. And one of them, while everything that was said correct, lacked a tremendous amount of detail and is only going to be worth 20, 30%. Right? And that's the problem. Sometimes people are losing points, not because they're getting information wrong, but because they're not giving enough detail. This class is detail specific. I want every intimate step, every immediate effect. So really, while you're describing these processes, think what happens next and describe that, and you'll get all your points. All right. Questions on that? All right, excellent. We have now described our first possible essay question, initiation of communication at the neuromuscular junction. But remember, as we also talked about, we've turned it on. we're starting the process. And even though our muscle hasn't contracted yet, we know this muscle action potential eventually is going to cause the muscle to contract, which means we also need to be able to turn it off. And that's the second possible essay question. Termination. How do we turn it off? All right. Now, let's think about this. How does it begin? I'm contracting a muscle and I decide to relax the muscle. Where does that begin?
In your head.
Yes.
Bingo.
Oh.
We make a decision. Absolutely, we make a decision. Now, in this case, when we decide to turn it off, remember, we have to produce action potentials to get it to contract. So if we decide to relax it, we stop generating muscle action potential, neural action potentials. So now there's no new positive signal spreading down the axon terminal. All right, no more big positive signal. And with no big positive signal, the cell goes back to its resting membrane potential. Oh, I need this to be smaller now, don't I? Yep. Back to. And if depolarization is getting more positive than the resting membrane potential, does anybody know the fancy term for what it means to go back to the resting membrane potential?
Hyperpolarization.
Close. Hyper is actually where you go below. In this case, we just want to go back to resting. So in this case, it's actually repolarization. Or, well, let's be consistent. Polarization. There you go. So repolarization, or to repolarize, means to go back to its resting membrane potential. Now, notice if we go back to, negative 70 millivolts, we're going to pass threshold on the way down. And when we pass threshold on the way down to the resting membrane potential, our calcium channels close. Oops. And if our calcium channels close, that means no new calcium can enter the synaptic end bulb. However, that's only half of our problem. Because remember, there's already a lot of calcium in there. If only there was a way for us to get calcium out of the cell.
The calcium pump.
Exactly. Now that the calcium channels are closed, now our pump can make a difference. Our pumps can now get rid of the calcium that is already in the synaptic end bulb. And so what happens is now there's no calcium. And if there's no calcium, there is no more exocytosis. So with no calcium, there is no exocytosis. No more exocytosis and no new acetylcholine enters the synaptic cleft. All right. So far, so good?
Yep.
Stunned silence means yes. Excellent. So notice we have a similar problem to what we just had before. there is no new acetylcholine entering the synaptic cleft, but we still have a lot of acetylcholine in the synaptic cleft. So now what we need to do is we need to remove the acetylcholine that is already in the synaptic cleft. And the good news is it turns out that there are 3 ways we can do this. All right, way 1. Guess what way 1 is?
Break it down.
Yeah, absolutely. We have Pac-Man there. Absolutely. So the first way is that enzyme, acetylcholinesterase. Esterase. That acetylcholinesterase is going to breaks it down. And if we break it down, it can't be used anymore. However, someone pointed out, we need to be able to release a lot of this acetylcholine to get all these muscles to contract. So absolutely, Pac-Man here is going to do his job of gobbling these things up. But we kind of want to reuse some of this stuff as well. So a second thing that can happen is some of this acetylcholine can be brought back, attached to the plasma membrane, or brought back in through channels, brought back into the cell, and stored inside the cell inside of a vesicle again. And when we take something from outside the cell and bring it inside, well, of course, that process is endocytosis. So with endocytosis, we can bring it back into the synaptic end bulb. But, and I think this kind of gets to some of the questions you guys were asking about before. Remember, this synaptic cleft is an open space. The motor end plate and the synaptic end bulb are very close to each other. The motor end plate has all those nooks and crannies to catch acetylcholine and keep it in this space. But is there anything really stopping the acetylcholine from actually leaving? Right? And you think about it, if an acetylcholine floats over to this space right here next to the muscle cell, is it going to have any effect on the muscle cell?
No.
No, because the channels are way back there. So the other thing that can actually happen to our acetylcholine is it can just simply diffuse away from the synaptic cleft. So using these 3 processes, we can remove all of the acetylcholine from the synaptic cleft. And if there's no more acetylcholine to bind, well, actually, let's just do it this way. Doop, doop, doop. If there is no more acetylcholine to bind to the synaptic cleft, no more, try that again. There's no more acetylcholine in the synaptic cleft to bind to the channels. Well, with no acetylcholine, our chemically gated cation channels close. So now these channels are closed. And of course, if these channels close, I'm running out of spaces to write this stuff. So let's sneak this up here. That there, and that there. Excellent. When our chemically gated cation channels close, there's no new sodium into the cell. Yes, also no potassium can leave, but remember, it's not really the potassium that's driving this. It's the sodium coming in. And with no new sodium coming in and things like the pumps kicking sodium out, and think about this, it's kicking out 3 sodium, which are positive, and only bringing 2 potassium, which are positive in. Notice every time it kicks that out, it also makes the cell more negative because 3 positive things leave, but only 2 positive things come in. So with no new sodium into the cell, then our motor end plate repolarizes. And if our motor end plate repolarizes, no new muscle action potential. Our muscle action potentials stop. And with no muscle action potentials, ultimately our muscle cell is going to relax. All right. Let's take a look at that. All right. Termination. Again, starts with the decision. When we make the decision, we stop producing neural action potentials and our neuron repolarizes. Calcium stops entering the cell and we get rid of the calcium that is there and we stop releasing acetylcholine. All right. We need to get rid of that acetylcholine that is in the synaptic cleft because the acetylcholine is what's causing the muscle action potentials. So some of it diffuses it away, some of it gets broken down by Pac-Man there, and some of it gets endocytosed. And once those channels close, we stop the muscle action potential. And that will lead to our muscle relaxing. So there you go. That is our second possible essay question. Termination of communication at the neuromuscular junction. Questions on that? Excellent. Stunned silences. I love it. All right. So we've learned how to turn it on. We've learned how to turn it off. To really see if we've understood how this system works, a great way to do that is to mess the system up. And the way we would mess the system up in this case is to modify communication at the neuromuscular junction. We're typically going to modify it using some type of toxin. And that toxin is going to disrupt the activity of this communication at the neuromuscular junction and have some effect on the muscle cell. Let's talk about one you guys might be somewhat familiar with. All right? Where might you find botulinum toxin?
In foods that were not preserved properly when they were
There you
canned.
go. Canned foods or jarred foods that have spoiled. Absolutely. Excellent, excellent, excellent. Right? That is absolutely one of the places where you would most likely find this botulinum toxin. And let's actually talk about what botulinum toxin does. Botulinum toxin is a bacterial toxin that is produced. And remember how I mentioned there are 25, so hold on. There, so botulinum toxin. There are 25 proteins that are used to make that vesicle move to the plasma membrane and release its contents. Well, it turns out botulinum toxin breaks just one, but one of the proteins that is used for exocytosis. It happens to be a protein called SNAP. You don't need to know that, but just know that it breaks one of those proteins. And what happens when it breaks one of those proteins is that when it breaks one of those proteins, even if calcium is present, no exocytosis can occur. We'll get to Botox in just a second, absolutely, but you're absolutely on the right idea. So what happens here is no exocytosis can occur. So let's walk ourselves through this process. I make the decision that I want to contract the muscle. I generate the neural action potential. The neural action potential goes to my synaptic M bulb. It opens up voltage-gated calcium channels. Calcium rushes into the cell, and when calcium rushes into the cell, what happens?
It reaches
Exocytosis.
or
Yeah, exocytosis.
Well, true. Normally, calcium comes in and causes exocytosis to occur, but my botulinum toxin has broken that protein. So even though there's a massive amount of calcium coming into the cell, can I release acetylcholine?
No.
no.
No. So even though I made the decision, even though calcium's entering the synaptic M bulb, no acetylcholine is released. And if there's no acetylcholine in the synaptic cleft, there's nothing to bind to the muscle cell. There's nothing to let sodium in. So does our muscle cell generate an action potential?
No.
No muscle action potential. And if there's no muscle action potential, can our muscle contract?
No.
Does that mean botulinum, whatever that bacteria was called, causes paralyzing?
Exactly. And not just a paralysis, but what we call a flaccid paralysis, where no contraction occurs. The muscle does not contract. The muscle is flaccid. Well, it doesn't necessarily kill you because, as a couple people pointed out, botulinum toxin sounds really gross. But if you give it a cutesy name like Botox, then people have no problem with you injecting it into their body. Heck, they'll have parties where they all get around and serve wine and inject it into each other's foreheads. Why do they inject it into each other's foreheads?
To get smarter.
No, I'm not
No,
sure.
to eliminate wrinkles.
Exactly. I've got all these pesky wrinkles on my forehead. And if I could paralyze these muscles so that there was no muscle tension in it at all, then my forehead would be super smooth. Yeah, I can't show emotions anymore, but as long as my forehead is smooth, that's all that matters. Because as we know, it's only the beauty on the outside that counts, right? Now, again, we jest, but you guys have also pointed out some very great examples of this. While cosmetically it is used to remove wrinkles, there are also some really, really important uses for it. There are certain types of migraines that are caused by a decrease in blood flow to the cranial region. And so what they can do is they will inject Botox into the blood vessels in the temporal region of the head, and that paralyzes the muscle so that the blood vessels dilate, more blood goes to that area. And again, it's not a cure-all for all types of migraines, but there are certain types of migraines that can be almost completely eliminated with the use of Botox, right? And again, someone mentioned armpits. Absolutely. Acetylcholine is also used to cause glands to release their sweat. So if we paralyze the nerves that we break the protein in the nerves so they can't release acetylcholine, your armpits don't produce sweat anymore. Yeah, you start to overheat and end up typically sweating more from other parts of your body to regulate your body temperature. But as long as you don't have that armpit sweat underneath your shirt, that's, again, all that matters. If you have an eye twitch or an eye turn, right? If you have if you're strabismic, then by putting Botox into the muscle, they can weaken the muscle to help with the eye turn, to realign the eyes and things along those lines, or overactive muscle activities and things along those lines as well. So again, absolutely. And that's one of the beauty of these things. Because we understand how botulinum toxin works, we can use these things to our benefit. Now, the problem, as someone mentioned, is that this is all great and fine and dandy until you really are at that Botox party, drinking lots of alcohol, and maybe they inject it a little too deep and it gets into your blood supply. And let's say it goes to a muscle like your diaphragm. What does your diaphragm do for you?
Helps you breathe.
It helps you breathe. Absolutely. It's your primary breathing muscle. Absolutely. And so if this Botox went to your diaphragm, suddenly you can't breathe anymore. Now, the good news is Botox doesn't last forever. The toxin gets in there, it breaks the proteins, but eventually the toxin degrades. Eventually a signal gets back to the neuron to say, hey, we've got a broken protein here. We can't release acetylcholine anymore. And our neuron can make more of that protein, ship that protein down the axon to the synaptic end bulb and fix it. But so again, those people who get the Botox in their forehead, do they get it once and then they're done forever?
No.
No, they have to do it every couple of months, 6 months, 3 months, 5 months, somewhere around those range, because eventually the neurons heal themselves. So the good news is eventually the neuron to your diaphragm would heal itself as well, but it could take 3 to 6 months. How long can
Wait,
you hold your breath for?
Not
Probably
that
not
long.
3 to
like
6
a couple
months.
minutes.
Yeah, probably not 3 to 6 months. Yeah, exactly. But again, it's fun to laugh and joke about these things, but that's the beauty of this. As we understand how these things work, and yes, there's some dangers involved with this, but as we understand how these toxins work, often these toxins can be used for important functions, or can be used for other types of positive things as well. So, like I said, the migraines, things along those lines, absolutely, and cosmetically as well.
You said
I don't think it would help with ADHD. Yes.
you said you have to wait for the neurons to heal, but don't you just have to wait for him to make the protein that the Botox broke down or whatever?
True, but remember, as long as the toxin's in there, even if you're making more proteins and sending it down, it'll just break it again. So you have to wait for the toxin to degenerate and go away. And remember, we talked about how, for instance, some of these neurons from the small of your back travel down to your big toe. They're a meter long. For some types of transportation, remember, way back here in the cell bodies where we make the protein, and then we have to ship it all the way to the end of the axon. And some of those transportation mechanisms can be as fast as 2 inches a day. So it can take a long time. You thought the UPS took a while to get your stuff there, right? We were complaining about the post office right before the election, but moving 2 inches a day can take a long time to get that protein down to your big toe. So it isn't. So repair from this is not a fast process.
So by getting a Botox, we don't damage the muscle cell, right? We just
Correct. Where you're not damaging the muscle cell at all. If you were to go in and stimulate it with an electron, it would contract. Right. So if you were to go in there and artificially stimulate the muscle can contract. It's just not able to because it's not getting the signal to tell it to contract.
Some people say you get more wrinkly after getting Botox. Is that true? Or does that has nothing to do with?
I haven't heard of that. I guess I've never heard of that before. I guess the only thing that I would think of off the top of my head is maybe with the muscle paralyzed, there is less movement of the skin. And so maybe some of the elasticity of the skin is lost from the lack of use. And so maybe when the muscle tone comes back, there's less elasticity to the skin. But I'm totally spitballing here. I've never heard of that before. I don't have any guess. I'm throwing out an educated guess.
Maybe we forget how bad our wrinkles look until they go away.
Maybe that's what it is. TMJ is another example of a place where, again, depending if there's too much muscle tightness, again, they can weaken the muscles by partially paralyzing them to help with that, help with grinding, things along those lines. Like I said, there are a lot of positive uses for Botox. And so again, it originally was a toxin. It is a toxin that comes basically from spoiled canned goods. Let's talk about another example. Another example of a toxin. Well, I don't have to worry about that. If I had a reduction, I wouldn't have any left. All right. Another toxin is curare. Any good adventure movie worth its salt. At some point, our protagonist and all of his native helpers are running through the jungle, carrying away the treasure that they have misappropriated from some indigenous population. And of course, all of the indigenous population are angry about this and so are shooting the blow darts at them. Those blow darts are, of course, hitting all of the helpers and knocking them out. But of course, our hero dodges it all and gets away safely. Well, the most common type of poison used in those types of blow darts and things like that are a toxin called curare. A curare can come from certain types of mushrooms, so there's actually some frogs that produce something very similar to curare. Curare is an interesting toxin in that what it does is it binds to and blocks the chemically gated cation channel. Oh yeah, I forgot the boulder rolling down the hill. Absolutely, right. It binds to and blocks the chemically gated cation channel. So basically what curare does is it comes in here and binds to our chemically gated cation channel so that acetylcholine cannot. It's like putting gum in the lock. We talk about how that acetylcholine is the key that turns the lock, but if you put gum in the lock, you can't get the key in there. So again, we make the decision, we produce the neural action potential, we open the voltage gated calcium channels, calcium rushes in, exocytosis occurs, but we can pour a whole bucket of acetylcholine into the synaptic cleft. And the problem here is the acetylcholine in the synaptic cleft cannot bind to the channels. And if it can't bind to the channels, yeah, it's a great way of describing it. It's a competitive inhibition, or what we call an antagonist. Cannot bind to the channels. And if the acetylcholine cannot bind to the channels, then no sodium into the cell, no muscle action potential. And so what kind of effect would that have? No contraction. And notice again, we get yet another flaccid paralysis. Coffee and nicotine are what we call, can be agonists. So an agonist is basically something that binds and activates the channel. And we're going to talk about those in just a minute. We'll get to that. Let's finish this one first. Notice this one is a similar effect to Botox in that once again, we get that flaccid paralysis. But notice one of the key differences with this one. In this one, no cell is damaged. Again, as we understand how these toxins work, we can use these things to our advantage. One of the things that you're seeing more and more of in medicine these days are more and more elaborate transplant surgeries, where they're transplanting hands, they're transplanting faces, all of these types of incredibly delicate work. Normally, one of the big challenges with this is that our muscles spontaneously, our neurons occasionally will spontaneously release a little bit of acetylcholine to produce muscle tone, right? If you're sitting in a chair like I am right now, and you reach down and touch your leg is what you would think of as being relaxed. But anybody ever felt the leg of a paraplegic before? Does their leg feel just like it's relaxed?
No, but I can imagine how it feels.
No, it is in that case, that's completely flaccid, like we were talking about, where there's absolutely no tone. It is just completely flaccid, you know, completely non-moving, no tension or tone to it at all. So we always have a little bit of tone. Yeah, a lot more squishy. That's a great description for it. Absolutely. Which is great. It's important. However, if you're trying to transplant somebody's face and you're trying to make the tiny connections between the blood vessels and the nerves and the muscles and the skin, can even a tiny change in tension in that area cause damage while you're trying to repair that tissue and transplant that tissue? Yeah, absolutely. So what they will do in the area that they're working, they will bathe that area in curare. And when they bathe that area in curare, it completely paralyzes that region that they're working on. They're able to make the connections that they want. And because this is in the synaptic cleft, this is outside of the cell and hasn't damaged it, then what they'll do is they will just rinse out the area. And when they rinse out the toxin, everything goes back to its normal function again. So again, another example of a flaccid paralysis, but notice this one doesn't damage the muscle cell or
Why would someone transplant their face?
Well, I think one of the first most famous case was a woman who was attacked. She was visiting a friend's house or something like that, and their friend had a pet monkey, and the monkey attacked her and basically ripped the skin off of her face. And so she was able to get a face transplant as a result of that. There was another face transplant that was just done within the last 6 months. I don't remember when it was. I believe that person was a burn victim. I don't remember exactly what happened, but I think there was a gentleman who just had a face, yeah, in August or something like that. I don't remember the specifics of it. Oh, is that what it was? Was it a attempted suicide? Was that the August one? Yeah, I had forgotten what the cause of for that one was, but yeah, there you go. So that was the more recent one. So again, those kind of things have happened. All right, one more way that we can modify this, and let me sneak this one in up here. So I think if I move that up there, and we sneak this over here, I can have enough room to talk about one more. Yeah, chimpanzee, there you go. All right. The last toxin I want to talk about is tetanus. Right. Where does tetanus toxin come from?
Not vaccinating.
Okay, you are correct in that you are more likely to be damaged by tetanus if you are not vaccinated, but the toxin itself actually comes typically from rusty metal, like stepping on a rusty nail or a rusty can or something along those lines, right? What tetanus does is similar to curare in that this toxin binds to the chemically gated cation channel. However, how tetanus is different from curare is it binds and activates it. So now when our curare comes in and binds to our acetylcholine channels, these acetylcholine channels open and they stay open because the tetanus won't let go. And so as a result, you have constant sodium entering into the cell. Yes, and constant potassium leaving, but remember it's the sodium coming in. So you have that constant sodium entering the cell. So our cell is constantly depolarized, and it produces a continuous action potential. And if this cell is constantly producing action potentials, what is this cell constantly doing?
Contracting.
Yeah, it's in a constant state of contraction, or what we call a rigid paralysis. Right? You still can't control the muscle, But in this case, it's in this contracted state where you can't get it to stop. You are correct. We do have tetanus vaccinations, which do help us to lessen the impact when you have it. Absolutely. But again, it doesn't stop you from getting tetanus. It just lessens the effect that the tetanus can have on it. And what is tetanus commonly known as? Yeah.
A disease.
Yeah, what's the common name for the tetanus toxin? If someone has tetanus toxin, what do they say that that person has? There you go, lockjaw, right? Because basically what happens is the muscle, like the jaws, lock up and they can't be relaxed. So you got that locked up muscle as a result of it. Yes, Maximilian. Oh, how does cerebral palsy affect the neuromuscular junction? So it doesn't affect the my understanding is that it doesn't affect the neuromuscular junction directly. What happens with cerebral palsy is you are getting inappropriate neural activation. As a result of that inappropriate neural activation in the brain, those signals get sent a lots of different places. And one of the places they get sent is obviously to the muscles. So when that occurs, that can cause, seizure-like symptoms where the person, muscles contract rapidly and things like that. It isn't because there's a problem or a damage to the neuromuscular junction, though. The problem is at that decision point. In this case, they're not deciding they want to contract the muscles. They're having inappropriate neural activity that causes uncontrolled and typically uncoordinated contractions of those muscles. So it doesn't, that also obviously affects muscle function, but not here at the neuromuscular junction. But yeah, it can be incredibly debilitating. All right. Yeah, if you look in some of the old textbooks at tetanus toxin, you see these people that are just, arched in these incredibly painful, rigid positions by this constant contraction of the muscles. Can be incredibly damaging and fatiguing. And think also, if someone, go back to those breathing muscles we were talking about. If someone has a flaccid paralysis and the muscles are all loose and flaccid, we can ventilate them pretty easily artificially. If everybody's respiratory muscles are all in a constant state of contraction, is it going to be as easy to ventilate that person and get their lungs to fill and empty in a way when it's like that? No, if they're in a contracted state, it's much, much more damaging when that's the case. Oh, wow, that is awesome. No, so Parkinson's. So again, all of these things, Parkinson's disease, even alcohol, these are definitely things that influence nervous tissue. But these are not things that are affecting directly the neuromuscular junction. And we'll actually talk about Parkinson's. We can talk about even alcohol and that kind of stuff when we get to the nervous system in the next section. So yes, those are all much higher order things where those problems are occurring and not directly related to the neuromuscular junction. All right, so there you go. So I told you we'd be layering layer upon layer upon layer of stuff. So here, all this simply put together is that first step. But if you think about it, as I mentioned, this is really 3 possible essay questions. We now know how to initiate communication at the neuromuscular junction. We now know how to terminate communication at the neuromuscular junction. And we now have some examples of how you would modify communication at the neuromuscular junction. Excellent. So 3 of our 7 essays that you're going to potentially be, not potentially, that I guarantee you'll be responsible for on the exam, we have now covered. All right. Questions on that? All right. That is all I want to do for lecture today. Oops, I wanted that instead. So what we will do, and all these pictures, I've taken screenshots of these pictures. I will post them onto our Canvas site. But again, you have that great video. You have all those other resources to make sure you're taking advantage of as well. All right, when we come back from this break, we are going to switch gears into our lab. We have a bit of an introduction into our lab. And then, like I said, for the rest of today after that, and for the rest of this section, we are going to focus on origins, insertions, and actions. So like I said, I strongly encourage you to have that Bones handout printed out. If you have the study guide with you, because I'm going to ask you the questions, and having the answers right there is definitely something that will be convenient to answer those questions. So have those out, and then and have a red pen and a blue pen. So get those things during the break, and then when we come back, we will dive into the lab stuff. So let's go ahead and go back to this page just because it's fun to look at. This is my favorite lecture. I love doing this on the big whiteboard. And back in ancient times when we had chalk, I love doing this when I have chalk because you would just be covered with chalk at the end of this. But here's all of that. And I guess there's a little room down here at the bottom to remind us. So let's come back at 2:45. And at 2:45, we will restart.
You are live during chalk times?
Yes, I loved chalk. Chalk. Yeah, when I was in grad school, at Davis, most of the classrooms still had chalkboards and chalk, and that tactile sensation of the chalk powder that you have on it. That's the greatest thing that I missed from grad school was the chalk. I loved writing on the board, and I loved playing with the chalk. So I'm a simple man with simple needs. What can I say? So Ariana, great question. Let's actually take a peek at that. So the two handouts you need, well, definitely need this one. Again, these are the bones. So that is the origin. This is where we're going to draw the origins and the insertions. And then of course, the other one is this one, the one that we looked at last time, the one that identifies the muscles you're responsible for, has the origins and insertions on it, and has the actions on it as well. So that when I ask you these questions, because when I ask you the origin of the masseter, two things could happen. You could either already have it memorized, or you can read it off the piece of paper. And so if you're active and read it off the piece of paper, then that would be a great way to take care of that. So yes, these are the handouts that you're going to need for that.
So we want to print them during break?
If you have, so again, having access to this, even if it's just on your phone or if you have an iPad or something, you want to look at that, that is fine. But as I mentioned in the last class, we are going to be drawing origins and insertions. So on this, I'm going to draw, because remember, you're going to have a skull on this exam, and I'm going to have an arrow pointing at this part of a skull. And I'm going to ask you to identify the muscle that inserts into this bone feature. And so having these drawn out is going to be a great way to study this. Could you be successful on this exam without it? Sure. Is it going to be a lot more work? Absolutely. So taking the time to draw these out, and remember, you need a red pen for the origins and a blue pen or pencil for the insertions, is going to be something that is going to be incredibly beneficial to you. But again, this is college, you're adults. If you don't want to do this, then you don't have to do this. That's the beauty of this. But most of you are spending a fair amount of money to be here with the motivation of being successful. So I would strongly encourage you to print these out and to have them, because I think it will help tremendously.
Thank you.
Yep. Again, you're doing this for your study purposes. So again, as long as you're consistent with your colors, I think that that would be fine. Just know that, like I said, by convention, and by convention, that means the rule that people have made up. So as you do web searches for these things or other things like that, just know that you're going to run into red. But yeah, I mean, you're not turning these in for homework. You're not turning these in for an assignment for me. This is a study guide for you. So yeah, if you want to use purple and green or orange and chartreuse or whatever, that's totally fine. Yeah, just make sure it makes sense to you.
One more time, which lab handout is it? One last time, please.
Again, this is called origins and insertions.
Perfect. Thank you.
This is this one, the origin insertion. And then of course, again, it would also be helpful to have the muscle anatomy list as well. All right. And again, remember, if all else fails, you can just click on them and see which handouts are there and pick the ones that you want and like. All right. Any other questions? All righty. We talked for a bit more, so let's go ahead and what time are we done? Yeah, we have enough time to do this. So let's go ahead and make it 2:50. All right. I'll see you guys in 15 minutes. We are done with the physiology for today. So we're on to the anatomy. And again, we've got that list of the muscles of the body that we are responsible for. You are responsible for all of the muscles that are on your list and all of the actions for the muscles that are on your list that are all listed there. So if a muscle has 2 or 3 or 4 actions, you are responsible for all of those. However, as we also talked about, all of the bold muscles that are on your list, you are also responsible for the attachment points. And when we talk about the attachment points, there are primarily 2, an origin and an insertion. So one of the key things to remember, and here we'll cheat and do a simple drawing. There's bone 1 and there's bone 2. When we have a muscle, that muscle typically is going to cross one or more joints. All right, if you think about it, if you had a muscle that attached from one part of one bone to a different part of that same bone, when that muscle contracted, nothing would happen. So that wouldn't be very, very useful. So typically they're going to cross one or more joint. And so as we talked about, when you talk about the action, you can talk about the joint that it affects. So again, this motion is a flex of my elbow. Or you could talk about the part of the body that moves. Because notice when I contract this muscle, what happens is the 2 bones don't come up and meet in the middle. Typically, what happens is one bone is stationary and one bone moves. So again, when I flex my elbow, my forearm moves, my upper arm stays stable. And so my radius and ulna would be the movable bone, and the stationary one would be my humerus. And that's kind of how we define origins and insertions. The origin, is, if I spell it right. The origin is the attachment that stays stable, whereas it is the insertion that is the attachment that moves. So when we identify origins and insertions, typically it is the fixed end that is the origin, and it is the movable end that is the insertion. And so it inserts on the movable. So again, as we talked about, I could say that this is a flex of the elbow, talking about the joint that moves, or I could talk about basically what the insertion is connected of. This is a flex of the forearm, the part of the body that moves. All right. Well, it totally depends on the muscle. Remember, we have over 600 muscles. Are there muscles that are going to originate from the femur and insert into the tibia or the fibula? Absolutely. But not all of the leg muscles. There will be other ones as well. So yes. So again, it would be, but if you're right, if it was one that flexed the knee, then likely that would be the type of attachment it would have. Now, again, this is a general rule. And of course, there are some other general rules as well. Origins typically are proximal, either closer to the core of the body or closer to the midline of the body. Origins typically are wider, so they often have a much bigger, broader attachment point, whereas an insertion typically is much, much smaller. And when we learn about muscles, one of the things that we'll learn about, one of the things we've talked about a couple times in this class already, is the biceps brachia. The biceps brachia is a 2 bellied or two-headed muscle. So we have these two bellies that sit side by side, and you are going to be responsible for identifying each one of them specifically. And typically what happens is each of these have their own unique origin. And typically what happens with a multi-headed muscle is their tendons come together into a single insertion. So again, when we talk about the bicep brachia, we are going to learn about the origin of the long head. We are going to learn about the origin of the short head, but they have a shared tendon and just one insertion. And then a muscle like the tricep brachia has three heads with three different origins, but still just one insertion. Yes, Laura.
So for, like you said, for those muscles with multiple heads, does the origin usually, is that usually going to be the proximal
Yeah,
part?
so yes, with a muscle, usually the origin is proximal, closer to the core of the body, or closer to the midline of the body. But again, typically means not always. And there are some pesky muscles that we'll talk about as well. One of them we'll talk about today is a muscle like the sternocleidomastoid, right? One of its functions is to flex the head, bring the head down, right? So obviously the part of the head that moves would be the insertion, and the part that stayed stable would be the origin. But we can do something interesting with the sternocleidomastoid. I can use other muscles to lock my head in place. And when I use those other muscles to lock my head in place and I contract my sternocleidomastoid, instead of my head going down, my chest comes up. So for some muscles, we can actually reverse the origin and the insertion when it's moving. So if you look at your list, you'll see the sternocleidomastoid both flexes the head, but it also elevates the sternum, elevates the rib cage. So like everything we talk about in this class, it's more complicated than that. But in general, and that's why we use the word typical, in general, these tend to be the case. All right. Questions on that? All right, the other thing, there's actually two more things that we need to talk about before we get started on the actual muscles itself. The first is this concept of a muscle group. The name like muscle group, not surprisingly, it is made of two or more muscles. After all, you can't have a group of one. But there are two other important criteria for a muscle group. The first is they must be similar in function. Notice I didn't say identical, but they have to be similar in function. And they must share at least one attachment point. So that means I have muscle 1, I have muscle 2, right? They must share an attachment point. Either the, no, don't want that to be pink. Either the origin or the insertion must be the same. The other one could be different, but they must share at least one attachment point. They must either have a similar origin or a similar insertion. This is important because in this class, you are going to be responsible for muscles and muscle groups. You will notice on the handout that there are some muscle groups that you only need to know the muscle group name. Take a look at your handout right now. And when you take a look at your handout right now, you will notice that there is a muscle group on the first page of your muscle list. Name the scalenes. The scalenes is actually made up of 3 different muscles. Does anybody know the name of the 3 different muscles that make up the scalenes?
No.
No. And do you care?
No.
No, because it's not on your list. So you are not going to be responsible for knowing the individual names of the individual muscles that makes up the scalenes. All right, they're anterior, posterior, and lateral, but we don't care. The zygomaticus is made up of a zygomaticus major and a zygomaticus minor. Do you care?
No.
No,
No.
because it's not on the list. So you just need to know them as a muscle group. All right. However, if instead you go to, let's start easy, page, what page is this? 4, the last page. You have a muscle group in your upper leg known as the hamstrings muscle group that is made of 3 individual muscles. Do you need to know the names of those 3 individual muscles?
Yeah.
Yeah, what are they?
Bicep femoris, semimembranous, and semitendinate.
There you go, exactly. So notice it's very clear on the handout. So many of your muscle groups, you just need to know the muscle group names. You don't need to know the individual muscles or the individual insertions or origins. You just need to know them collectively as one muscle. So we will talk about those muscle groups basically as a single muscle. However, like the quadricep femoris, like the hamstrings, like the rotator cuff, there will be some muscle groups that you will need to both know them as a group, but you will also be responsible for the individual muscles that make them up as well. All right. Questions on that? All right, excellent. And again, we'll go through this list together. We'll make sure that we emphasize that as we go through that. The last thing when identifying the muscles is there are basically 3 main factors. Do I have that listed here? Nope, I don't. 3 main factors that are used in determining the names of muscles. One is the region of the body. So for instance, the bicep brachia is in the brachial region of the arm, where someone just mentioned the bicep femoris is in the femoral region of the arm. So the region of the body is one of the ways that we identify muscles. The action of the muscle is one of the ways we name them. So guess what a flexor muscle does?
Flexes.
Guess what an adductor muscle does?
Adducts.
Adducts, exactly. So, right, an elevator elevates. So again, they're often named by their functions. And the third thing that they're named by is their shape. Obviously, the bicep femoris, bicep brachia has 2 heads, so it's shaped that way. But other muscles are named based on their shapes, and that shape is determined by their fascicles. Remember, we talked about the organization of a muscle. We know that individual muscle cells are bundled together into a fascicle. So again, that perimysium, that dense irregular connective tissue, bundles up the muscle cells into a fascicle. And all of the muscle cells in a fascicle are parallel. Right? This is convenient because if muscle cell A contracts or muscle cell B contracts or muscle cell C contracts, this fascicle is basically going to do the exact same thing. It's going to get shorter in this direction. Right? So all the muscle cells in a fascicle are parallel, so they all contract in the same direction. But the same thing is not true for fascicles. Not all fascicles are parallel. So one fascicle in a muscle could be like this one here, but a second fascicle in a muscle could be oriented like this. So obviously, if this fascicle or any of its muscle cells contract, or if this fascicle and all of its muscles contract, you are going to get a different action of these muscles. So the orientation of the fascicles determines their shape, and notice are also going to affect their function. All right. So again, all muscle cells in a fascicle are parallel, but all fascicles are not. So it is the organization of the fascicles that determines the shape of the muscle, and that influences the function. And as you can see here from this simple illustration, there are 4 major shapes. So let's draw these. I find this easier, again, if we simplify things a little bit and draw it. So 4 main shapes for muscles. I don't know why this has to be pink. The first of these is what is known as parallel. And again, guess how the fascicles are oriented in a parallel muscle?
Straight.
There you go. Exactly. Fascicles are parallel. Absolutely. This happens to be the most common type of muscle that we have in the body. And I'm not going to bother drawing the fancy circular ends or anything along those lines. But again, here we have 3 fascicles, all parallel to each other. Notice when we wrap this up with the epimysium, we typically get that spindle shape where we have a tapered end with the 2 tendons coming off of the end, and then this kind of spindle-shaped muscle with the muscle in between. Obviously, the advantage of this is all of the fibers are pulling in the same direction. So we have tremendous coordination in this. And let's think about this. When our muscle cells contract, as we know, they're going to get shorter. But as they shorten, they also thicken as well, right? So notice as this muscle contracts, what happens is the muscle cells get shorter, but they also get thicker as well. So as the muscle shortens, it bulges. Again, you may not have thought of it in these terms, but you're aware of it. Because what is the primary reason to work out? Why do we work out? Why do we use our muscles?
Lose weight.
Say again?
Lose weight.
Okay, make money to get bigger muscles. But true, you want to get bigger muscles. What's the advantage of having bigger muscles?
To be stronger and healthier?
True. Okay, healthy. Yeah, sure. Everybody cares about healthy. No, nobody cares about healthy. It's all about wooing women, right? The whole reason to work out is to woo women, right? Exactly. You go onto a campus, and when you go onto that campus, you walk up to some big buff guy and ask him where the library is, right? Hey, Laura's got it. Absolutely. You ask that guy where the library is. Oh, the library? Oh, it's right over there, showing off the guns as he's showing you where to go to. Absolutely. Right. So it's all about the wooing of the women. So you get that bulk of the muscles when you contract it.
Like, yeah, man, check out my pecs.
Exactly. Right. So exactly. The whole point is when these muscles contract, they get shorter as a response to that. So again, it provides great coordination. However, the one downside, if there is such a downside to something like this, is again, it only pulls in one direction. There are times when we're going to want to have more mobility to a muscle. And if we want more mobility to a muscle, we can do that with a different shape of muscle. Oh, sorry. Hold on, where'd that go? Oh, there it is, okay. Hold on. What we call a convergent muscle. With a convergent muscle, basically the fascicles are arranged in a fan shape. So basically, and in this case, I'm just going to draw them as lines rather than boxes. So a fascicle would be oriented this way, and another one this way, and another one this way, and another one this way, and so on and so forth. Notice what you get with this type of a muscle is a big, broad, spread out attachment on one side, where it comes together basically to a single attachment in the other side. And we were just joking about the pectoralis major muscles, right? What's a good exercise to work out your pectoralis major muscles?
Push-ups.
Chest exercises?
Yeah, bench press, like a bench press, absolutely. However, or flies, those kind of things. If you're doing flies or bench press or something along those lines, do you always want to do it laying completely parallel to the ground and just do it in the one direction?
No.
No, if you really want to maximize the gain in the pectoral region, you also have to do it declined where you're pushing up, or you have to do it inclined where you're pushing down, right? You want to go the full range of motion for it because, again, it's on the shoulder and it allows for a wide range of motions. So with this one, the advantage of this convergent fan-like appearance is it gives a lot of range and diversity to the movement. All right, the next one, our third type, what are called pennate muscles. Yes, Laura, you had a question?
Yeah, sorry, really quick. So the convergent or the fan ones, are those like specifically for kind of like your abdomen area where it's kind of more defined, or?
So in this case, it can be in that region, but I think it's more going to be associated with your shoulder and your hip, right? Those are the joints that have the widest range of motion. So those are the ones where you're going to want much more diversity in the orientation of the fascicles so that you have that ability to not just pull the arm across, but to pull it up, to pull it down, and so forth. So you're going to get more range of motion. So I think these are going to be actually more associated with the shoulders and the hips. All right. Excellent. Pennate muscles, basically the key to a pennate muscle is, again, we have parallel fascicles. But not parallel to the tendon. So in this case, for instance, we have a tendon, a single tendon oriented like this. And what'll happen is that all of the muscle cells will come into it at the exact same angle. So notice all of the muscle cells are going to be parallel, all the fascicles are going to be parallel to each other, but not parallel to the tendon. This gets its name pennate because it kind of has a quill-like appearance to it. Quadriceps femoris is an example of this. The rectus femoris of the quadricep is one of the muscles that is like this, yes. Although not this particular one. We'll get to that one in just a second, but you absolutely have the right idea and why this is so important. Why is this orientation so advantageous, do you think? Well, let's think of it. Go ahead. Yeah, strength, exactly, right? If we collectively were playing tug of war, and all of you were on one side and all of I was on the other side, and we were playing tug of war, who would win? You guys or me?
Trevor is heavier.
I'm sorry?
Us.
Yeah, exactly. You guys would win. Unless we were using a 3-inch rope. If we were using a 3-inch rope, would all of you be able to get your hands onto that 3-inch rope?
No.
No, and so that's not necessarily going to give you an advantage anymore. However, if all of you are able to get your hands on that rope, you guys are going to be able to generate a tremendous amount of force. And that's what's happening here. This allows us to get lots of fascicles attached to a single tendon. This can give us muscles that are relatively small in size, but very, very strong. This is the kind of thing that is incredibly useful in your forearm, right? Unless you want Popeye forearms, you wouldn't necessarily want parallel muscles in this region. We want to have small muscles in that small compartment, but we want to have that strong grip strength. And so having that strong grip strength comes from having all of these parallel fascicles connecting to a single tendon. Now, this particular type of pennate is what is known as a unipennate, where it's one line of fascicles and one tendon. Well, weight definitely is an advantage in tug of war because obviously the other team has to move it and you know, being able to move it, but it's also very much about leverage and force, how much force you can generate as well. Right? If you doesn't matter how heavy the team is, if the whole team can only get one finger on that rope, then you're not going to win, no matter how heavy you are. Right? So again, it's also about being able to generate that force. All right. A second type of pennate muscle also has a single tendon, and it has two rows of fascicles. Trying to be consistent with my coloring here. There you go. One row of parallel fascicles on one side and one row of parallel fascicles, on the other. The best example of this, as someone mentioned earlier, is your rectus femoris. Your rectus femoris is one of the most powerful muscles in your body. It isn't necessarily the biggest, but it's one of the most powerful because its job is to move that big, huge hunk of meat that is your leg and flap it around through space, right? Your leg is a big, huge, heavy piece of meat. If you want to quickly lose 20 pounds, cut off a leg, right? And so to be able to move that around, we need a big, strong, powerful muscle. And so that rectus femoris, that straight muscle in the front of your leg, has two rows of tendons, two rows of fascicles connecting to that single tendon that allows you to swing that with a tremendous amount of force and energy. And this is, we call a bipennate. The last type is what we call a multipennate. A multipennate actually has two or more tendons to it. Ugh, it's supposed to be round. So multiple tendons with multiple rows of fascicles. So I'll only draw a couple here in the interest of time. So a couple on this one and a couple on this one, so you get the idea. And I got the pretty pictures from your textbook that we can look at for this as well. So again, we have multiple tendons with multiples, and this gives us both strength and flexibility of movement in a small compartment. And again, probably the classic example of this is your deltoid muscle. Your deltoid muscle up here in your arm, we need to be able to bring it up and move it around. So again, the bipennate was the rectus femoris. The multipennate is that deltoid. All right. Excellent. So those are all great examples of those. Our fourth type of muscle, the arrangement of the fascicles, is a circular muscle. And guess how the fascicles are arranged in a circular muscle?
Circular.
Yeah, circular arrangement. So we have the fascicles around in a circular arrangement, absolutely. And the advantage of circular muscles, when you construct a circular muscle, what happens to the lumen, the space in between?
It closes?
Yeah, it gets smaller. You've got the right idea. When it contracts, that lumen gets smaller. However, you absolutely have the right idea. If we were to have a circular muscle, absolutely, orbicularis oculi, orbicularis oris are both examples of that. If we had a circular muscle that was large enough that when it constricted, not only did it decrease the size, but it actually closed the opening and acted as a valve, what would we call that fancy circular muscle that acted as a valve? Come on, I know you know. Well, we probably don't want an artery to close, but what would we call a valve-like muscle? No, not the cardiac valve. How about a sphincter? There we go. Excellent. We call it a sphincter. Oh, I misspelled it. Yep, sphincter. Absolutely. So
Wait, this is something. Do we call the lumen a sphincter or?
No, the lumen is the gap inside of a circular muscle. Is the hole, the opening in it. If our circular muscle, the muscle was thick enough that when it contracted, it closed the lumen entirely, so that the lumen was closed, so that it acted like a valve and could control the movement of stuff going through it, then we would call it a sphincter. Okay. And again, if you don't like my pretty pictures, as we've talked about, we have them here. Here we see that parallel muscle that, again, when it contracts, gets shorter. Has that spindle shape, typically with the two attachment points on the end. Here we see that big, broad, fan-shaped convergent muscle. Again, that allows much more flexibility of movement. Here we see the pennate, feather shape, our unipennate, lots of fascicles on a single tendon. We get a lot of strength in a small compartment. And again, here we see that bipennate, single tendon, parallel fascicles. Here we see the multipennate. Notice when you look at that deltoid, at first it looks a little bit like a convergent, but as you see in the cross section, there are actually multiple tendons that the fascicles are all connected to. And then lastly, that circular muscle, which, as we said, when you contract the lumen, the space inside of it gets smaller. All right. Questions on that?
So when the lumen closes, that's when it forms the sphincter.
If not all, so not all circular muscles are thick enough where they can actually completely close the opening. So all circular muscles will control the diameter of the lumen, making it bigger and wider. So all circular muscles.
But if it can close, it would do that.
Yeah, but if it's thick enough so that it can actually close, then it would be a sphincter. So
Okay.
all sphincters are circular muscles, but not all circular muscles are sphincters. Does that make sense?
Yeah, it does.
Excellent. All right. With that, we are done with our introduction. And again, our goal is now going to talk about origins, insertions, and actions. And the one last thing I want to remind you about before we get to this is, once again, to beat yet another dead horse, to encourage you guys to take advantage of the resources that are available to you. Because again, the other resource that I'm hoping most of you used when we were doing the bones was your practice anatomy lab. Right, this had those bones in them that were great. It had lots of quizzes and tests that you could use for that, which is great. And again, we can use this for the muscular system as well. Now, it is cool to look at the muscles on the cadaver and see the individual muscles and stuff like that way. It can be fun that way as well. However, that's not how you're going to be tested. You're going to be tested on the models. But conveniently enough, there are models here as well. So for instance, as I said, you can look at head models that show the muscles and stuff like that. So we were just talking about the orbicularis oris and the orbicularis oculi, which should be there. We could turn the labels on where it shows all of the different muscles and stuff that we can hide. Oh, it doesn't label the orbicularis oculi. Interesting. But it probably does on a different picture as well. So we have all of these, use these as well. However, that I think I've showed you as well. The other thing I think I've showed you, but I want to remind you about as well, not only do you want to come to the anatomical models to see all the different muscles, like not just the head, but the arms and the legs and all of that, but notice up here are animations. When you click on animations, and for instance, you go to the upper limb, we can take a look at that bicep brachia muscle.
Biceps brachia
Can you guys hear that?
Yeah.
Yeah.
is a large two-headed flexor muscle located in the anterior compartment of the arm. The origin of the long head is from the supraglenoid tubercle of the scapula. The origin of the short head is from the tip of the coracoid process of the scapula. The two heads insert by a common tendon onto the radial tuberosity and by the bicipital aponeurosis, which blends with the deep fascia on the medial aspect of the forearm. Biceps brachii flexes the forearm at the elbow supinates the forearm, and weakly flexes the shoulder. It is innervated by the muscular cutaneous nerve.
What I love about this is she does give a little bit of extra information that you don't necessarily need to know, but all the important stuff is definitely here. I love how they show the muscles in isolation. I love how they show us the origins and the insertions using that red and blue like we expect. So we see them on the bones, we see the joints that it crosses, and then it also shows us the actions of that muscle. So again, if you're having a hard time with these muscles, these resources that you have online, along with the stuff that we're going to go over together, is something that should help tremendously. All right. So again, if you're not using your resources, shame on you, but now's the time to start. All right. So let's start going through our list together. And again, to try to help to compartmentalize this and make a little bit more sense, make the information more easy to digest, kind of just like we did with the bones. We did the bones of the skull. We did the bones of the vertebral column. We did the bones of the arm. We're going to do the same thing here. And we're going to start with the bones, with the muscles that move the head and the neck. The first muscle, as we can see here on the, and again, I'll tell you right now that all of these pictures are from your textbook or from previous versions of your textbook, which means your textbook has a similar picture to them here. We have this superficial cheek muscle here known as the masseter. All right. So if we were to go to our handout, and let's zoom in a little bit. Excellent. And I wanted to draw my origin and my insertion. What is the origin of the masseter muscle?
The zygomatic arch.
Excellent. So notice I can take my red pen or my red pencil, and I think I need a thicker line to make this a little easier. And I can make that red. Excellent. And what is the insertion of the masseter?
The mandibular angle.
There we go. Now, like I said, obviously one of the reasons we're doing this is because there's going to be a skull on the exam, and I could have either of these bone features labeled and ask you to identify the muscle that originates from the zygomatic arch, or I could ask the one for the inserts into the mandibular angle, and you need to know that. But the other advantage of taking the time to do this is, again, remember when we see the attachment points, we can see the bones that, or in the joints that are being crossed. Notice in this case, it's the jaw, that temporal mandibular joint that we were talking about earlier. And remember one other thing. When a muscle contracts, it pulls the insertion towards the origin. So notice by just looking at the origin and the insertion and thinking of the action that it would be, what is the one action that the masseter would do?
Evaluate mandible.
Right. Elevate the mandible, absolutely. Bring the mandible up. When would you do something like that?
Eat and talk.
When we're eating cookies at your house.
There you go, exactly. You guys and the cookies. Absolutely. Although be careful with chewing and speaking, because again, especially chewing, I think like, you know, the whole cud-like action. But this is definitely the start of it, that first bite. That bringing that jaw up for that first bite. Yes, this is going to be used in chewing and talking as well. But really, if you really want to think about just what it does, this is just bringing the jaw up for that very first bite, that elevation of the mandible. Notice this is important, and we'll go back to the picture in just a second, but I want to do this while they're here, and I can, because if I leave the screen, the drawings won't be here when I come back. What's the origin of the temporalis muscle?
Temporal fossa.
Yeah, so notice it's this, right, that smooth surface of the temporal bone right here. And what's its insertion?
Coronoid process of mandible.
Yeah, remember that's basically this here. Now notice the zygomatic arch blocks it a little bit. So remember, you're not turning these in for a grade. So do this however you want to do it. If you want to draw it there as close as you can see it, that'd be fine. If you want to do it as a dotted line or change the color to remind you that it's going to be up in here a little bit, it's hidden a little bit by this part. Again, the whole point of this is for you to see it, for you to be able to visualize this. And notice in this case, once again, when we contract, it is going to pull the insertion towards the origin. And notice this time, it's not bringing it straight up. So notice when we contract this muscle, it's doing not one, but two actions. Obviously, like the masseter, it brings it up. And what would bringing the jaw up be called again?
Elevation.
Yep, elevate the mandible. But what else does it do to the mandible?
Retracts it.
Retracts it, brings it back. So notice, by taking the time to draw these two origins and insertions, we can see these muscles that are similar in their location and similar in their function, but we can also see the differences between them as well. Now, as you guys absolutely pointed out, these are used in chewing, these are used in talking, and in fact, you can experience this for yourself. If you put your hand on the side of your face and you clench your teeth, you should be able to feel both the masseter and the temporalis muscle contracting. You can feel that change in tension in the temporalis and in the belly of the masseter in those two regions when you clench your teeth. So when you clench your teeth, you should be able to feel those as you're doing that. All right. And so again, if we go back to the pretty picture here, we see this here. And that brings me to the last thing that I wanted to talk. Actually, that's not true. One more thing I need to talk about out of this. The second to last thing that I want to talk about for these. So on this exam, you are responsible for names, origins, insertions, and actions. And when a muscle is like the masseter, that it only has one action to it, it's usually pretty easy to understand and remember. But as you start to get to 2 actions and 3 actions, and some of our muscles will have 4 actions, it can be tricky to remember all of them. So one of the things that I'm going to have you do, and again, I expect you to do this individually, but if it becomes an issue, then I will divide you guys up into groups and we'll do this as a graded assignment, but I'm hoping you will actively participate on this on your own. What I want you guys to do is to come up with some mnemonics to help us to be able to remember these things, right? So for instance, the temporalis helps you to both elevate and retract your mandible. And when would you do that? When would you both elevate and retract your mandible? Can anybody think of an example of when you might do something like that?
Would that also be chewing?
True, absolutely chewing. But again, my problem with the terms like chewing or talking is that they're relatively vague terms, right? There really doesn't describe an actual action to that. So you're absolutely correct. These things are used for chewing and talking. But is there any time where you would just elevate and retract your mandible? I can tell there aren't too many older siblings out there, because as an older sibling myself, anytime my younger siblings did anything even remotely stupid, which was quite frankly most of the time, right? I would have to give them that face, that dork face, right? And how do you do that dork face? You give that big exaggerated overbite, right? So you elevate and retract your mandible. And when you do that, you can do that nice big, that durr, right, for them for when you're elevating and retracting it. So that durr face that you give them when they're being a dork is a great example of the type of activity that would involve that temporalis muscle. All right. So starting next week, I'm going to start expecting you guys to start to come up with some of these tricks to help us to remember it and to share them with the class. So when I ask for someone like that, someone will turn on their camera and someone will demonstrate their durr face or whatever it is that they can think of to help us to remember that. And then we'll all laugh and we'll move on to the next one. All right. So like I said, I'm encouraging you to do this because it's going to help. A, it's going to help you study, and B, this is part of your muscle assignment. If you've looked at your muscle activity handout, that's basically what the muscle activity handout is. I give you an example of either a common activity and you have to tell me what muscle that does it, or I give you the muscle and its actions and you come up with a common or not so common. I'm not sure the durr face is a common activity, but something that can help us to remember it. Let's see if we can come up with some other examples. Here's a fun one. Well, but you at least have to stand up and do it. But yes, it is hard to coordinate, but as long as people are participating, then I won't put you in groups and force you. If I have to put you in groups and assign the muscles and force you guys to do it, it's not as much fun and you don't get as much out of it. All right. Sternocleidomastoid. We have muscles that, like most of the things that we run into in the body, are big alphabet soup terms. But often when we have these big alphabet soup terms, they tell you everything you need to know about it. This muscle has 3 attachment points. So let's switch to our drawing and see if we can figure out what the 3 attachments of the sternocleidomastoid might be. What do you think the first attachment of the sternocleidomastoid would be?
The sternum, the manubrium.
Sternum, absolutely. So it attaches up here to the manubrium of the sternum. What do you think clido refers to?
Clavicle.
Clavicle. Notice also, this is not a continuous attachment. It's not like it attaches all the way across. There's going to be some muscles that are like that. But in this case, these are 2 separate attachment points. So we have an origin that starts on the sternum. We have an attachment point that starts on the superior surface, medial or sternal end, superior surface of the clavicle. And what is its insertion?
The mastoid process.
Mastoid process of the temporal bone. Now notice I can do this two ways. If I want, I can cheat and I can draw it over here, because notice over here next to it, we can actually see that bump behind the ear. Notice this picture doesn't really give us the bump behind the ear. So I could draw an arrow and do it this way if I wanted to do it that way. Or honestly, what I do is I just cheat and I draw my own mastoid process. I just put my own mastoid process out here. There you go. So again, you're not turning these in. So I want you to do on these sheets what is going to help you to be successful. There's no wrong answers as long as it makes sense to you and it helps you to learn this material. Questions on its origins and its insertions? Let's take a look at it here. Notice this picture, it shows it covered up by other muscles. So notice if we peel away some of those other muscles, we get a much better sense of what that sternocleidomastoid is. And again, notice we can clearly see here, it's attaching to the sternum, it's attaching to the clavicle, and it's attaching to the mastoid process of the temporal bone. So questions on its attachments?
So the sternocleidomastoid attaches to the sternum and the medial surface of the clavicle, correct?
Yep. Yep.
Okay.
Or the sternal end of the clavicle, right? However you want to describe that is fine.
And then when you were talking about we were gonna like come up with ways to like memorize it. Is that next lecture that we have to do that, or?
Yes, so starting with the trunk and with the arms and the legs, I want you moving forward. I will give you the ones that I have here, but if you happen to have a good one for the sternocleidomastoid, I'm happy to hear it. Yes? Well, let's figure out its actions first. What are the 3 actions of the sternocleidomastoid?
Deflex the head and elevate the sternum.
Excellent. So let's start with those 2. Notice, like most of our muscles, this is a paired muscle, meaning we have one on the left and we have one on the right. Our origins are down here. Our insertion is up here. So when I contract both muscles at the same time, and actually let's stop looking at the picture. When I contract both muscles at the same time, it brings the head down. It flexes the head. In fact, this muscle is sometimes referred to as the prayer muscle, because it's the muscle that would lower your head in prayer. However, remember, this was the example of the muscle that I used, where if I use other muscles to stabilize my head, when I contract this, instead of my head coming down, my sternum and my clavicle come up. So basically my rib cage comes up. All right, so if I use both, I can bring the head down or I can bring the ribs up. However, notice if I just use one, if I just contract one, I bring this one mastoid process towards my clavicle, and what it does is it rotates my head. Notice if I contract the other one, it rotates the head the other way. So notice if I just use one, I can rotate my head. If I use both, I can flex my head, or I can elevate my rib cage. So those are the 3 actions. Can anybody think of something you might use this for?
Nodding your head.
Okay, sure. So absolutely nodding your head is one of it, although that doesn't really put the rotation into it or the elevation, right? When we're thinking of one of these activities, you want to make sure it includes all of the actions. Ariana, do you have one? Looked like you were talking, but you were muted.
No, like what about like when you do yoga, you know how like when you start to like do like the yoga your techniques, you kind of like rotate your head a little bit.
Yeah, when you're stretching your neck for rotations, that might be, you're taking the deep breathing. I like that. Yoga might be a good example. Circle head banging for Metallica would be a good example. I like all of those, absolutely. Those are all great, really, really good, but I will be honest with you, they're not my favorite. My favorite happened about, gosh, at this point, like 10 years ago, 12 years ago, And what I might point out was before it was legal. But I had this student, because again, in class, I actually assign this on the first day. And as groups, I make you actually stand up and demonstrate. And I had this student who sat in the back of the class, was all quiet, very demure, kind of like Madison, just sat all quietly and was demure in the back of the class. And she stood up to demonstrate, her words, not mine, what she called a bong rip, right? Flex the head, elevate the rib cage, rotate the head. So there you go. There are your 3 actions that you put together and give you the actions of the sternocleidomastoid. So if you can remember how to do a bong rip, you can remember the 3 actions of the
Beautiful.
sternocleidomastoid.
Chef's kiss.
So there you go. Exactly. All right. Excellent. So, and that's what I'm looking for. You know, we laugh at that. That's funny, but hopefully now none of you will get that wrong on the exam because you will all easily, hopefully be able to remember that. All right, excellent. So there are 3 actions for that. And yep, 2 more. Excellent. With that, where are we on time? We're doing good. Perfect. Oops, nope, wanted to share that. Here is a second view of the sternocleidomastoid. Again, notice we are seeing how it is attaching to the mastoid process, to the sternum, and to the clavicle. And again, remember, it's paired, it's on both sides. But they've removed it on the other side, so we can see our first muscle group. This first muscle group, as I mentioned, is the scalenes. And as I mentioned, it's an anterior, posterior, and middle, but you don't need to know those. You just need to know them collectively as the scalenes. So again, I do not care if you know the individual names of them. It does not matter. We are going to talk about them collectively as a group.
A quick question.
Yes.
So the sternocleidomastoid is made up of scalenes, or it's a different muscle we're talking about now?
Scalenes is a completely different muscle.
Okay.
They are similar in their locations. So as we'll see, they're similar in their actions. But these are different muscles, just like the masseter and temporalis. Different muscles, different actions, but similar locations and similar functions. Notice both of them brought the jaw up. These are 2 different muscles, similar locations, but different origins, different insertions. So they are going to have different actions. So let's draw them. Where is on my picture here? Is it here? Scalenes, there you go. It's on this one here. All right, so again, for ease for me, because the way I have this organized, I'll just cheat and put it over this one where the sternocleidomastoid is, but you can draw it over here next to it. All right, so what is the origin of the scalenes muscles?
The transverse process?
Of?
The C2, C7?
Yeah, C2, C3, C4, C5, C6, all the way to C7. Notice my picture doesn't show all of them, but I can draw enough of them here to be able to get the idea of it. And what is its insertion?
Skin and muscles. Oh wait, no, anterior, lateral, surface of ribs 1 and 2.
So anterior and lateral surfaces of ribs 1 and 2. So basically, this is a muscle down in this region. Now, notice, like the sternocleidomastoid, it is similar in its location. So again, it can bring the head down, flexing the head, or it can elevate the ribs. However, notice this one goes from the neck to out here on the lateral part of the ribs. So when it contracts, when we just contract one of them, rather than turning my head to the side, it pulls my head to the side for a lateral flexion. So notice I can flex the head, I can laterally flex the head, or I can elevate the rib cage. So 3 actions for our scalenes? When might you use that?
Also during yoga.
Okay, yoga is a good example. This is usually what's happening in the classroom right now. I usually point out because one or two people in the classroom right now are kind of like, huh? When they're looking at this, and that's the one I always think of. I always think of like, you know, that confused dog staring at the vitriola when it's playing the, you know, the animals sound. I always kind of think of that, like, you know, kind of flex a little bit, you turn your head to the side in confusion, maybe sigh real deeply because you're confused. So I just think of that as kind of that confused look. So that's the one that I kind of think of when I think of the scalenes. Excellent. All right. Notice there's only one more muscle we need to know the origin and the insertion of. What is it?
The zygomaticus.
Zygomaticus. Notice the zygomaticus again is a muscle group made up of two muscles, the zygomaticus major and the zygomaticus minor. But again, I don't care about that. We are going to just talk about them collectively as the muscle group. So you just need to know them collectively as the zygomaticus, period. Excellent. So what is the origin of the zygomaticus?
The zygomatic bone.
Yeah, notice not over here on the arch. We're talking about the apple of your cheek. And what's the insertion?
The skin and muscles at the corner of the mouth.
Right. Remember when we talked about skeletal muscle, we said most skeletal muscle connected to bone, bone to bone. But here's an example of one that doesn't. This one, obviously this picture doesn't have skin or muscles on it, but we can cheat a little bit and draw the corner of the mouth here where the skin and the muscles are. So it attaches to the lateral aspect of the mouth. Of course, as we know, when a muscle contracts, the insertion is pulled towards the origin. So when you contract this muscle, it elevates the lateral corners of your mouth. And when might you do something like that?
Would it be smiling?
Yeah. How about when the class is over, right? Then try to take the smile off your face at that point. Absolutely. So elevating the corners of the mouth, which you would use when you were smiling. And there you go. Just that easily, we have identified the muscles, the origins, the insertions, and the actions of all the muscles that you're responsible for that are bolded on the head. Now notice, there are still some other head and neck muscles we need to talk about, but you don't need to know the origins and insertions. And remember, it doesn't mean they don't have origins and insertions. They absolutely positively do, but you don't have to worry about them. You just need to know, identify them, and you need to be able to know their actions. So let's go through those. Starting first with a deep muscle located here in the cheek region, which isn't surprising since this cheek region is called the buccal region. Deep to that is the buccinator. And notice it's a deep muscle. It's, well, is that other picture? Yeah, this is nice. Okay, excellent. It's a deep muscle that sometimes can be tricky to find if you don't know what you're looking for. But notice you take the zygomaticus that comes down this way. You take the masseter, which is coming down this way. And notice in that pocket in between, that is where you see the buccinator. So that buccinator is basically between the zygomaticus and the masseter. But it is a deep muscle helping to form the walls of your cheeks. So that's pretty simple and straightforward. And here we see that nice deep muscle there as well. Since it's helping to form your cheeks, what do your cheeks do for you?
They smile.
True. Okay. Although I would argue that the smile is more in the lips and definitely in the eyes, but they can play a part in that. What else do the cheeks do for you? Yeah, one of their main jobs is to contract and squeeze to present food to the teeth for chewing or mastication. So their job is to squeeze inward to be able to squeeze inward to present that food to the teeth for chewing. And that's its primary function, right? What's a good activity that would help us to be able to remember that? When do you
Taking a snack during break.
Okay, when you're taking a snack during break, absolutely, you're gonna have to use it there, right? When you're squeezing the cheeks, right? Although there's also chewing and a whole bunch of other stuff that go on with that, so you got to be careful with that stuff as well, right? You could think of it in terms of pursing your lips to like whistle or blow a kiss, but remember, you're also doing that with your mouth as well. So eating a lollipop is good as well. However, when I think of the buccinator muscle, it of course reminds me of the greatest movie in the history of all mankind. Fish Face is getting you closer. What, of course, is the greatest greatest movie in the history of all mankind? How about Zoolander?
I'm dead.
There you go. Blue Steel. Right? Notice with Blue Steel, he is using his buccinator to enhance his cheeks, right, for Blue Steel. So there you go. So this is that Blue Steel muscle that we use for that. All right, excellent. Let's talk about the next muscle. Our first circular muscle, the orbicularis oris. What is its job?
To close and protrude the lips.
Yeah, close and protrude the lips. So when you're whistling, when you're blowing a kiss, something along those lines, it protrudes the muscles for that. Notice it's not the only circular muscle on the face. We also have the orbicularis oculi. What does it do?
It blinks.
Well, we want to be careful with blinking, right? Because when you blink, you're just bringing the eyelids down. Notice this is a circular muscle. So remember, with a circular muscle, we want to squeeze. So this isn't so much as the blink as it is, you can think of it in terms of the wink, or right, if you're going out and it's really sunny outside, or if it's really dusty outside and you're kind of squeezing your eyes closed, it's more of that physical squeezing of the eye closed, not just the blink, but the squeezing of the eye around it. You have levators of the eyelids that are what cause the eyelid to go up and down. So this is really about squeezing your eyes. Yeah, when you get that nightmare and you're squeezing your eyes closed at night to do that, absolutely. Right, or your wife's trying to talk to you and you're trying to pretend you're asleep. Yeah, exactly. All of those things, great examples of when you would squeeze those eyes shut. Absolutely. And one last one. And this brings us to the last, let's switch to this picture. The last rule we need to know for this exam. There are, as I pointed out and mentioned before, muscles with multiple heads or bellies. Again, they can be called heads, they can be called bellies. Like the bicep brachia, like the tricep brachia, like this muscle, the occipital frontalis. On the exam, I'm going to have this picture with a big, huge arrow pointing right here, and I'm going to ask you to identify this muscle. And on the exam, if you say occipital frontalis, you will get it wrong. Because what I want is what this specifically is. This question here with this arrow and this question here with this arrow are two different answers. This one over here on the left is the occipital belly of the occipital frontalis, and this one over here is the frontal belly of the occipital frontalis. And I will give you a big, huge, fat warning that this is the kind of answer I am looking for. On the exam, what you will see is you will see it phrased exactly like this. Be specific. If you see those magical words, be specific, let warning bells go off. Because that is my way of telling you that I want a precise piece of information. Right? These two arrows are not the same answer. Identify the muscle, be specific. One is the frontal belly of the occipital frontalis, and the other is the occipital belly of the occipital frontalis. Don't just say occipital frontalis. Don't just say frontal belly and occipital belly of the occipital frontalis. If I give, and again, I'll just give you one arrow. So if I give you one arrow, I want that precise thing that is, and I want you to be precise. And again, I will give you those two words that the warning bells should go off majorly. If you see the words be specific, then you know I am looking for a very precise piece of information. In this case, the precise belly. And as I also mentioned, when we have multiple bellies, they have also multiple origins. So if I was at this one, you don't need to know the origins for, but when we get to the bicep brachia, for instance, you need to know the long head, you need to know the short head, and you need to know theirs specifically as well. So when I say be specific, I'm either going to be asking for a name of a muscle or an origin of a muscle, and I want it for just that one specific head. Okay? Yep, that is why we learn the bones. And again, frontal bone, occipital frontalis. Absolutely. It tells you, it's another one of those, an alphabet soup term, which tells you everything about it. It's the occipital frontalis, right? It's got two bellies, one in the front, one in the back. What does it do? What does the occipital frontalis muscle do?
Elevates the eyebrows.
Elevates the eyebrows, absolutely, right? So again, that ability to elevate the eyebrows, raise the eyebrows. So then we don't want to use raise, we want to use appropriate anatomical terms. Elevate the eyebrows, like when you're being surprised or something. And notice something else. Notice at the bottom of your list, there is a structure you are responsible for. A structure is something that is not a muscle. In this case, notice there is a big, broad, flat tendon that connects the two bellies of this muscle together. As we know, a big, broad, flat tendon is an aponeurosis. This one happens to be on the top of the head, so it's the epicranial aponeurosis. Yeah, you can't do it with Botox, the cheesy guy in the movie. I told you it was all about women. I'll show you that in just a second. So there you go. This structure on top of the head is the epicranial aponeurosis. And if, like me, you conveniently don't have a lot of hair, when you use your occipital frontalis, not only do you see the eyebrows move up, but you can see the whole scalp move. Right? You can see that whole scalp going back and forth because it's all connected by that epicranial aponeurosis. See, there are some advantages to not having hair. All right, questions on that? Excellent. And as you mentioned, notice, well, there's one more muscle we need to talk about in a second. All right, let's do that last muscle. Then I'll remind you of what we were saying before. All right, one more muscle that you are responsible for, and that is the platysma. Notice most of the muscles we've talked about have started from the head and come down. What's interesting about the platysma is this big, huge, broad, flat muscle that basically sits over the top of your neck. It's a big superficial muscle. You're not going to see it on most of the models and most of the pictures because if they leave it there, you can't see any of the muscles underneath. But it's this big, huge superficial muscle that drapes over the neck and, as you notice, comes all the way up onto the lower jaw, on close to the cheek, which is where we get its actions. Notice its actions. And what else do I have? I've got one here. There's something else here. Oh, here we go. Excellent. This is the chart in our classroom, which normally we would have to see, we'd get to see. And notice again, here you see this big, broad, flat muscle that is very superficial right on the front. And that is that platysma. And you can see how it comes up towards the lip, which again helps you to understand its function. While you don't need to know its origins and insertions, looking at it can help you to understand what it does. It is going to bring the jaw down, depress the mandible, and because it comes onto the front, it can also pull down and protrude the lower lip. All right, giving you that pouty lip and causing your jaw to depress.
Can you show it again on this slide? I actually turned away, so I didn't catch it.
Here it is on the slide.
Okay, what about the next picture?
Oh, this one. Oh, I see. It's, hold on. This muscle right here, this drapey muscle right here on the neck.
Okay, thanks.
Yeah, down there. Excellent. All right, so there you go.
So help us in bringing our head up to look up like this guy is doing, or no?
No. And in fact, notice as he's bringing his head up, he's stretching the platysma out. The platysma, when it contracts, brings it down. This is another one of those muscles. And again, I've been letting my beard grow, so I don't know how well this will work. But because this is a big superficial muscle, this is another one of those muscles where if I hold my jaw in place and I contract this muscle, I can put tension in the platysma and you get that Hulk neck, right? That Hulk neck that sticks out, right? From that if you feel that when you do that, that muscle that you feel sticking out, that is that superficial platysma that you're doing that way. And like I said, as you guys were pointed out, the cheesy rom-com, all of these movies, right? As we talked about, all of these muscles we use to woo women, right? You use your orbicularis oris and your buccinator, right? You guys were talking about at the beach or at the seashore. You know, you're at the beach, you see that cute girl across the way, so you whistle at her or you blow her a kiss. You waggle the eyebrows at her, you give her the big exaggerated wink, and then when she still turns around and walks away, you're sad and you're angry, right, as a result of it. So there you go, using all of those muscles that we have talked about and identified. All right. Excellent. And that's how the game's played. We are going to continue to go through our list, identifying muscles, identifying origins, identifying insertions, identifying actions, and coming up with goopy things that help us to remember them. I've sat here for the class for an hour now and made a fool of myself doing these silly things. I would like some help with these silly things as well. So not only, again, because you're getting graded on it as part of an assignment that you're going to be doing, so you should be motivated to do this, but it helps to break up the monotony of the class as well. So anything you guys can add to it, I'll continue to add mine that I have, but like I said, most of the ones that I have are things that I've stolen from students because students are just absolutely hilarious when they come up with these things. So I think they're awesome. So any that you guys want to add to this, that you're bold and brave enough to demonstrate for us, we would all greatly appreciate. Exactly. Yeah, that again, like I said, normally with jokes, I will, like, if I get it from a student or something like that, I'll credit them for the first couple of years. And at that point, I figure it's mine. But like I said, it's been 10, 12 years or something like that. And I just was so blown away by that student. I was in tears laughing. It was the best thing that's ever happened in the class. That was so hilarious. But so I will continue to use that one forever and give her credit. Because I just think that it like you guys said, it crystallizes it so perfectly. It's the perfect example of how of those three actions and how you would use them. And it just at the time, because again, now marijuana use is much more common now than it was then, and so it's just that much funnier back then. So it just was quite humorous. But anyway, so there you go. And like again, the point, of course, is that it's nice that it's funny. It's fun to be silly. But if it helps you to remember, then that's really the point. And so hopefully by being silly, by talking about these things like that, it helps you to remember these things. These are mnemonics. Like on the exam, I'm not going to ask you what muscle allows you to do a bong rip, right? But if I will ask you what the three actions of the sternocleidomastoid are. So if you can remember the three actions of the sternocleidomastoid, because you can remember a bong rip, then you'll get that right on the exam. All right. So what we did today is what we're going to do in the next 4 lectures, 5, 2 weeks worth of lectures, where you're going to do physiology during lecture, and then we're going to do origins, insertions, and actions during lab. So that is the game plan for the next, well, with the holiday, practically the next month. All right. Questions on any of that?
I have a question, but
Yes.
only because I saw that you have your 5-for-5 midterm meeting.
Yes,
Are
thank
they
you.
open or?
So no, so
all
again,
closed?
my goal is always to get the tests back in 1 week. But between having tests in both classes and some of the other craziness that is going on right now, I was not able to complete them yet. Once I get the tests turned back to you, we will be over the halfway point, and it is a good opportunity, like we did in the first week of class, to get together again and do a 5-for-5. So I want to encourage students to come to the 5-for-5 to talk about things that can help them to either maintain their grade or improve their grade, and as a carrot to encourage people to come and take advantage of this. If you have a C, D, or F in the class after the next exam, I will actually give you 10 points of extra credit if you come to my office hours to discuss your grade with me and how you can improve it. If you're getting an A or getting a B, I still want you to come. You can still get your 5 points of extra credit, but I really want to help the people who are struggling to figure out what they can do to help them to be successful. Hopefully what we did today in lecture also, where we really talked about how I'm looking for step-by-step details on the essay questions. Hopefully, if you take that to heart and practice that, that can be something that can help you to be successful as well. But I think it's a good point to check in with people. So yes, it is on the schedule right now, but I hadn't talked about it yet, and I hadn't set it up because I typically wait till after the grades are done. So I will definitely have the grades back to you on Tuesday when we meet next. Again, as soon as I'm done graded, I'll get them posted, and then you'll get to see them on Tuesday. But then also on Tuesday, after Tuesday, I will open up the some slots where we're able to meet and do the 5-for-5. So yes, that is something that we will be able to do, but I want to do it after this last test has been graded. All right. So thank you for asking that. But yes, we will get there. There isn't anything you have to worry about that yet. We'll start worrying about that next week. All right. Any other questions? All right. Awesome. You guys have an excellent day. And today's Thursday. You have an excellent weekend. Study hard, and I will see you on Tuesday.
Have a good weekend.
All right. Bye, guys.